it can take an average of 10 to 15 years. So it's a very long and expensive endeavor. Now, we're going to take each of these components, drug discovery and preclinical part of development. Then we'll progress into the clinical phase, which is the phase involved with human testing before you can get to register a product. And it's been licensed for use in actual patients. Now, let's start from the research phase, the discovery phase of drug development. In this phase, that's the laboratory component of drug development. Most of these happen in the context of a laboratory. And it's a very important first step because you have to understand and identify. Please go on mute. I will appreciate that. So drug discovery, I think we can all relate with depending on where you are from. A lot of people here share my background. Yeah, so malaria is a very common tropical disease. And growing up in Nigeria, most of us are familiar with a lot of local remedy where people take different kind of plants that have been known for their medicinal value. And what they do is attempt to extract the active component of the medicinal plant and boil the plant, and you take a cup of it, and that's your medication for malaria. Yeah, but in the 1970s, a plant known as Artemisia annua has been identified as having active ingredients that works against malaria. So scientists try to develop this, extract the active ingredient to develop into a drug known as artemisinin. And even fast forward to today, there are a number of artemisinin combination therapies that are known to treat malaria, and that's in fact the standard of care. But the knowledge of Artemisia annua, it's mostly what is passed on, what our great-great-grandfather know about plant, about medicinal property of the plant, and that's passed along through the generations. Yeah. Today, knowledge has evolved. We know a lot more about disease process. We know a lot more. In fact, in certain disease, specific genes has been implicated in the disease process. So it's beyond what is passed along through the generations. Knowledge has evolved. Drugs discovery today is then beyond just knowing about a particular plant. In fact, what is done today, technology, where scientists in the laboratory are able to screen literally hundreds of thousands of compounds at the same time, trying to identify compounds that have potential to have activity to impact the disease process for a given disease. And in the laboratory where that is done, again, that involves thousands of compounds. And when you are doing that, you can imagine the number of statistical issues that can come up in that context. One is the nature of the experimentation itself. where you are doing large number of tests and trying to identify compounds that have potential hit against the disease, the target. So you're generating heaps of data. And this is prone to false positive. You may have some things that appears to be hit, but they're not necessarily true. So false positive is something you have to contend with. And even the setup of the experiment itself, it also presents different experimental design issues because there are a number of factors at play in the setup of the experiment. The role of positive control. When I say positive control, there might be a compound that are already demonstrated and validated to have activity against the disease. So incorporating positive control helps you to understand what is real and what is not. Then you can also have negative control. So these are different aspects that present experimental design issues that statistical thinking can help to address. Moving on, when a compound has been identified and optimized to be moved forward in development against a particular disease, we don't just move from the laboratory piece that I just briefly described into clinical trials. It doesn't work like that. There has to be a lot, extensive preclinical testing in animals to ensure that, in fact, it is safe to proceed into human. And I will share an example that even illustrates this or underscore the importance of preclinical testing as we move along. But preclinical testing includes in vitro, what is called in vitro testing in the laboratory, again, where you isolate cell and you try to study impact of the compound on isolated cells. And then you proceed into what is called in vivo testing, where you are actually administering the compound in animals. And the focus of this is to understand the toxicity profile of the compound. And this by itself would take several years because depending on the disease, depending on what is known about the compound, there are different experiments, different tests that needs to be conducted. An example that underscores the importance of preclinical testing is what is commonly known as the thalidomide disaster. This is something that happened in the late 1950s or early 1960s. A German drug developer introduced a drug known as thalidomide into the European market. And it was introduced as a sedative. It is used in people with anxiety, people with sleeping disorder. And eventually, its use was extended to treating sleeping morning sickness in pregnant women. And it was this extension in usage that resulted into what is now known as the thalidomide disaster. And it was recorded that about more than 10,000 of children were born with different kind of deformities, as this picture I'm showing depicts. Yeah, and at that time, it's because of the limitation, the limited knowledge of statistics, the limited knowledge of toxicity itself, these different kind of studies that were conducted. In particular here, the gap happened to be that there was no study conducted in pregnant animals to understand the effect of the drug in fetus development, how the drug can impact the development of an embryo, yeah, before it was introduced into human. If that were to have been done, the potential effects, the potential toxicity could have been highlighted before it was introduced into humans. And this is very real. Even today, there are a number of survivors of this thalidomide disaster.
Good luck, say you did church. I say.
I'm going to try and mute. Well, I don't have that functionality. So please, if you can go on mute, we will greatly appreciate it. In response to the thalidomide disaster, naturally, regulators respond to this. There are a number of regulations introduced to avert recurrence of this type of disaster. So this particular example is just to illustrate the importance of preclinical testing. So after extensive preclinical testing of a compound, and if the developer at that point believe that appropriate evidence have been generated to establish that this could potentially be safe in humans, you then put what is known as investigational new drug application together. That's what is called in the US here, for example. It might be labeled differently in other jurisdictions, but it's a dossier that's essentially compile all your evidence of preclinical testing to date, and you submit it to the appropriate regulatory authority. Ask for permission to proceed into human testing. When you move into human testing, the starting phase of that is the phase one of clinical trials. In phase one, usually this is the first time that you are administering the drug in humans. So it's very carefully done. It's also often the case that this is not done in actual patients. And the reason it's not done in actual patients is because you want to be able to understand what is the effect of the drug as opposed to the effect of the disease. So you don't want the two to be confounded. So that's why it's often done in healthy volunteers. And there are exceptions to that, especially in oncology, where most of the compounds are very toxic. And because of the life-threatening nature of cancer, in that case, it's often the case that even phase one studies are done in real patients, not healthy volunteers. In other areas, it's usually healthy volunteers. But the focus in human testing is safety, to begin to understand the safety profile of this compound in humans. There you want to understand the pharmacokinetics and the pharmacodynamics of the drug. That's a way to say that how is the drug processing, how is the body processing the drug? How is it absorbed into the body? How is it distributed? How is it metabolized in the body? And finally, how is it excreted from the body? And you also want to understand what the drug itself is doing to the body. And as part of the need for carefulness, it's often very sequential. where you start with a single dose in ascending order of increasing dose. So you do a very small cohort of patients, say 3 patients to start with, in a very low concentration of the drug. If it's proven to have an acceptable safety profile in that cohort, you progress to the next higher dose, and so on. And then you move into multiple dosing, where multiple dose of the drug can be administered to a subject. And one of the key objectives of preclinical or of phase 1 clinical trials is to establish what is known as the maximum tolerable dose. What do we mean by the maximum tolerable dose? It's the maximum dose of a compound that does not cause unacceptable side effects. So usually, you will define what is known as dose-limiting toxicity events. These are events that are labeled as a toxic effect of the treatment. And from your phase 1 testing, what you capture is the data that shows the proportion of dose-limiting toxicity events with the different dose levels. And at the end, you have a profile, something like this curve I'm showing here, that gives you the probability of dose-limiting toxicity at each dose level, okay? And usually you would define what is known as, what is an acceptable probability of dose-limiting toxicity. So for example, if 33% or 0.33 is used as your target toxicity limit, the objective then is to find the maximum dose that has a probability of dose toxicity limit below 0.33. And you use the curve to establish that. And that's how you arrive at your MTD. Once the maximum dose, the maximum tolerable dose is established, the next phase of development is the phase 2 development. In phase 2, you are beginning to move into patients to begin to understand the effect of the drug in diseased patients. It's the first set of evidence that you begin to generate that this drug, it actually has the potential to treat the patient that is targeted to treat. And we call this proof of concept, often labeled as phase 2A. And in phase 2A, what you are trying to establish, again, is a proof of concept. Usually, you have real patients involved, say 50 to 200 patients over several months of treatment, depending on the disease. And once that is, once proof of concept is established, you move into those finding studies. I will speak a little more about that. But for the proof of concepts, a lot of time we use Bayesian methodology to establish proof of concept. Typically, you will have 2 criteria. The first one is to establish that the posterior probability of the treatment the effect parameter being less than the minimum threshold is given the data is very small. In other words, the predictive probability, the posterior probability of the treatment effects measured by a defined parameter exceeding the minimum threshold is high. That means the converse of this probability criteria that is shown here. That's the minimum. And then the second criteria that is often relevant is that this particular effect that you are demonstrating is clinically relevant. So in that case, a second threshold will be defined. And your objective then is to show that with some decent posterior probability, given the data, you can establish that the posterior probability of the treatment effect parameter exceed that target threshold. Once proof of concept is demonstrated, the next step is phase 2B, where you're looking at a range of doses. And what you want to do here is to characterize the dose response profile of the compound with respect to efficacy, but not just efficacy, with safety as well, whereby you are looking at different doses and looking at the level of response for efficacy. Then you look at the same for toxicity or safety. Very often in most compounds, you have a monotonic dose response profile, whereby with increasing doses of the compound, you have increased efficacy response, okay? And but the relationship is often the opposite, right? Between efficacy and safety. As you increase the concentration, you are more likely to get increasing efficacy, but decreasing probability of the dose being tolerable as you increase on the dose scale. So it's a careful balance between those two, so that you are defining a threshold where you are achieving the acceptable defined clinical relevant effect, but also defining the maximum dose that is giving you an acceptable safety profile. If they define as dose with acceptable safety profile is higher than the dose giving you an acceptable efficacy, then you have what is known as a therapeutic window. Yeah, it's within that window that we're going to select a dose that we're going to move into further development. The next step of development is phase 3 testing, which is often labeled as confirmatory testing phase. Again, in confirmatory testing, you are testing in real patients. The target population where the drug is intended for. And this is where you put together everything that you've learned in the development process in phase 1, in phase 2, and this is the most rigorous phase of clinical development because it's the basis of approval that regulatory authority will look for. And in different jurisdictions, there are different requirements. But in terms of the number of patients that are involved in the study, later on in the course of this workshop, my colleague, Dr. Smith, he will talk about how we size this type of studies. And it's depending on the disease, depending on the magnitude of effect that is expected. The range can be very broad, from hundreds to thousands of patients. the focus is often, or let me say the requirement, often it depends on the jurisdictions, but in most jurisdictions, you are required to have a study that you have a predefined statistical test criteria, a type 1 error, or significance level that is not more than 2.5% one-sided, yeah? And also, in confirmatory studies, you can have one dose. In some cases, you have multiple dose, but usually not more than 2, usually. Again, in the US, typically you will require 2 independent phase 3 studies. In some other jurisdictions, that could be different. There are jurisdictions that will allow you to do 1 study. For example, China or Japan. In some cases, once you've completed your phase 3 studies, as part of the phase 3 program, maybe additional safety study might be required. It's often the case that maybe in a particular disease context, the duration of the study that is required for efficacy is different from what is required to establish safety. So you can, additional safety study might also be required. In addition to evaluating efficacy in phase 3, evaluating safety is a critical component of it, yeah? So the objective, again, efficacy and safety of the patient. And when it comes to safety evaluation, it's not just one parameter or one endpoint we're looking at. It's often multi-dimensional, where you're looking at different aspects of safety. But very commonly, yes, you're following, you are capturing data on adverse events that occur in the course of the study. In some disease area, you are capturing specific laboratory parameters that are targeted to be indicators of safety issues. You're watching vital signs, you're watching electrocardiography of the patient. So these are different types of data points that might be relevant for safety evaluation. But also, often, safety, there's a lot of subjective expert review involved in safety evaluation. And unlike efficacy assessment, where it's often driven by hypothesis testing, a lot of time for safety, it's often descriptive, where you are not performing hypothesis testing, really. You are just analyzing the overall totality of the safety evidence accumulated across the different parameters. When phase 3 studies are concluded, and at that point, the sponsors still believe that the efficacy and safety of the compound are sufficient to justify moving forward. The next step is to submit a new drug application. So the new drug application will be submitted to the responsible regulatory authority. In the US here is the Food and Drug Administration. In Nigeria is the NAFDAC, yeah? So you collect the dossier up to that point, and it's submitted for review. That can take time as well. In the US, that can take 10 to 12 months on the average to review the package. And at that point, if the regulators agree with the sponsor that sufficient evidence has been generated to demonstrate the safety and efficacy of the compound in the target disease population. The compound is then granted license to go into marketing. But that's not the end of it. Even after securing an approval, the job is still not done. In some cases, there are still gaps in our knowledge. There might be gaps in safety still. Maybe we don't know the long-term effect of the study. There are studies that can take years. We want to see, for one example that comes to mind is a gene therapy, where regulators, after approval, requires that even the patient that were used in phase 3 study, they followed up for 15 years so they can understand the long-term effect of the drug on these patients. So there's post-marketing activities. You can have post-marketing commitment studies. And one other example that I want to quickly illustrate here is a phase 4 example of a drug known as Bios. Bios was approved in the US in 1999 to treat arthritis and acute pain. And following its approval, there was a post-approval study commitment that the sponsor was mandated to conduct. And it was in this post-approval study that additional evidence was generated showing that the drug is actually presenting heart attack and stroke risk to patients, especially at the high dose of usage. And in 2004, the drug was eventually withdrawn from the market due to the additional evidence, post-marketing evidence that was generated. So in summary, drug development, very complex process, high risk, prone to failure. You can, in fact, for the sponsor, it's often beneficial if you can fail faster, if you can fail at the earlier phase of it, because as you move along, it's very expensive. So to fail at the latter phase can be very disastrous. It has huge financial and other implications for patients and also for the sponsor. So phase 1 development, you start small. It often starts in healthy volunteers where you are attempting to understand the pharmacokinetic and pharmacodynamics profile of the drug. And you move into medium-sized study in real patients to establish proof of concept and identify the dose that you can move forward into further testing. Phase 3 is a confirmatory testing phase. And then it's often the case that you have phase 4 activities, post-marketing activities, engendered to generate additional evidence for the drug. It's also in that phase that you maybe look into indication expansion. You want to test into another disease population or test in some other sub population than previously tested in earlier phases. So there are a number of statistical challenges as you move through the development spectrum, and different challenges provide opportunity for different statistical applications. We will move on to highlight some of these applications as we move forward in the course of this workshop. Moving on to principles of clinical trials. In principles of clinical trials, we'll be focusing on the clinical phases of clinical development. clinical phase 1, phase 2, up to phase 3. However, most of the principles I will be talking about it applies across, but it's often much more emphasized in phase 3 development because that's the confirmatory phase. And as I said, that often serves as the basis for regulatory approval. So what is a clinical trial to start with? A clinical trial is an experiment in humans. It's an experiment where you are studying the effect of the drug. You're providing that as an intervention to humans. And it's also a designed or planned experiment as opposed to something that it's happening naturally in nature and you are just observing. There are a number of ethical safeguards that has been developed over the years, usually in response to disasters. For example, the one I talked about earlier on with respect to thalidomide. There's what is known as Declaration of Helsinki, which was introduced in 1964. And there are two basic requirements of Helsinki. One is that you need independent ethics committee approval for clinical studies, independent of the sponsor of the study. It also stipulates that the participants in clinical trials have to be consented. They have to voluntarily agree to participate in the experiment. It's not acceptable for a patient to go to a doctor's office and unknown to them, they're actually being used as part of an experiment. That's not acceptable according to the Declaration of Helsinki. There are other regulatory oversight and guidance that are often provided by different regulatory authorities. And in addition to that, there's also what is known as the ICH, International Council of Harmonization. And most of the principles I will talk about today are actually addressed in what is known as ICH E9, which speaks to statistical principles in clinical trials. Now, when we talk about clinical trials, there might be a misconception that when you are doing this experiment, you get all the hundreds or thousands of patients assembled and at once you administer the treatment and at once, at the same time, you do the efficacy and safety assessment, you capture all the data concurrently at once. No, it takes time and it starts from a careful design, careful planning of the study, putting concepts together. What is the question that we want to address? What is, you define the target population, right? You define the assessment that needs to be conducted as part of the study to address the clinical question of interest. And you proceed to write a clinical study protocol. A clinical study protocol is like your plan, a detailed plan that instructs the investigators, the clinicians that are actually interacting with the patient. It's like a procedure for them, telling them what is the objective, what are the assessments that needs to be conducted, at what time do they need to conduct this assessment, right? It also stipulates the analysis that is going to be done to address the objective of the study. And not only that, you have, in most cases, an independent document called the Statistical Analysis Plan that often has to be finalized before you even enlist or enroll a patient in the study. So when all this is done, you enroll the first patient, and that milestone is usually labeled first patient, first visit. And it will take a while to accumulate all the targeted number of patients that you want to enroll in the study. So following a period of recruitment, you enroll the last patient in the study. And then if the period of observation from the first treatment to the end of treatment is say 3 months. So 3 months after the last patient first visit, you have last patient last visit. And then after that, there's a number of operational activities, cleaning the database, ensuring that there are no database issues that needs to be corrected. So all this is done between the time you have the last patient last visit and the time of the clinical database log. The clinical database log is when all the data have been captured, it's been adjudged to be clean. At that point, no more data entry is going on. We freeze the database. At that point, it's also often the case that the studies are blinded or masked, meaning that in most cases, you don't know what the patient are receiving. And I will speak more to this later on. And then you start the analysis after database log towards generating what is known as a clinical study report. So you see, it's a number of steps, starting from designing the study, the actual conduct of the study, the execution, and moving on to reporting phase. Yeah, so the involvement of a statistician is not just at the reporting phase. The statistician is an integral part of defining the questions that need to be answered, how data will be captured, designing the study, and all through every stage of the process. This is a particular example of a clinical trial in multiple sclerosis. Multiple sclerosis is a disease of the central nervous system, and it's manifest with different signs and symptoms. But very common in multiple sclerosis is what they call relapsing and remitting multiple sclerosis, where the patient have a period of time that they have a disease episode. And after that period, the things stabilize again. That's the remitting phase. But the relapse is a recurring process. So you have multiple episodes of the relapse. And the objective of this study is to generate evidence that the drug is reducing the relapse rate after 24 months of treatment. And the comparator in this case is a placebo. So you are comparing the new drug, actually two dose level of the drug, to a placebo. The design is a parallel design. What do we mean by parallel design? This is a design where you have different cohorts that you are administering different treatment as part of the study. So in this case, 780 patients were enrolled and randomized into 3 different groups. Group A receiving a higher dose of the compound, Group B receiving the lower dose, and Group C receiving placebo. So placebo is the control group. The question that will come to mind here, why do we even need a control? So that's one of the principles that we're going to be talking about. We're also going to touch later on how the sample size is determined. One element here that is also important is randomization. Why is randomization relevant? Why should we randomize? Why can't we use other methods of assigning patients to these 3 treatments? So these are the principles that we'll touch on very briefly today: control, randomization, blinding, and lastly, the statistical analysis method itself. It's placed in the protocol. So control group. This is to say that a group of patients in the study will be receiving a relevant alternative treatment from the drug that is of interest. In most cases, the alternative treatment is either a placebo, which means that there's no active substance of the drug in the makeup of what is being administered as placebo. Or in some other cases, you have the current standard of care being administered. And we compare what happens in patients receiving the drug to the control. And the rationale for doing that is because we want to establish that the difference we're seeing between the drug and placebo is actually the effect of the drug. Because if you don't have control, you can do like a before and after treatment comparison, okay? If you do a before and after treatment comparison, what you might be seeing, it can just be reflecting a phenomenon known as regression to the mean, whereby, let's say in the multiple sclerosis example that we're talking about, a patient came into the study when they actually have an episode, and maybe the episode is pronounced after some time, but naturally, the patients have a period of time where they are not having so much episode. So that natural phenomenon itself can manifest as a treatment effect, and we don't want that. We don't want regression to the mean to be represented as a treatment effect. So we want to be sure that what we are seeing is the effect of the drug. So we want to compare the drug to a control. And in most cases, it's a concurrent control, meaning that in the same study, under the same experimental condition, you are observing both the group that is receiving the drug and the group receiving control. And in some exceptional situation, you have a control group that is external to the current study. You're using historical data. If you are still here on Wednesday, we will have a presentation by Professor Nikki Best on the use of external control. So stay tuned for that. The other element that we've talked about here is the aspect of randomization, where you are randomizing patients into the different groups. And usually the way you do that is through a computer-generated list where you generate a sequence of numbers that correspond to the different groups in random order. In most cases, you're doing one-to-one randomization, but in some cases, it can be unequal randomization. But also, let's take, for example, if you're going to enroll 50 patients into a study, we don't just randomize the sequence of 50 in a single block. We want to ensure that as we move along in the course of the study, at small intervals, you can ensure that you don't deviate too much from the targeted randomization ratio. So we define a randomization block. So it can be a size of 4. For example, if you are doing one-to-one randomization, the trick is that the block size has to divide evenly into the sum of the randomization ratio. Yeah, and we also have sometimes a stratified randomization. I will speak a little more to that as we move along. In some more advanced cases, I spoke already about the possibility of unequal randomization, but you can have response adaptive randomization, where as you move along in the course of the study, you are modifying the randomization ratio based on how patients are responding to the drug. Why is randomization necessary at all? Randomization is necessary to prevent bias. At the end of the study, we want to be able to make a conclusive determination about the effect of the drug. We don't want a situation where physicians have, because of the profile of the patient before them, they want to do what is best naturally for the patient. And so when they see somebody that is sicker than some other patient, there's a tendency that they want that patient to receive an active drug, an active treatment. Yeah. If that is done, at the end, the effect of the severity of the treatment may be confounded by the effect of the drug. So we want to ensure there's a balance. So we don't want subjective assignment where physicians are assigning the drug based on what they want to do for the patient or based on what they think, based on how they think the patient might respond to the drug. So to avoid confounding effect, we randomize. Also, randomization is used to ensure that what we're seeing is at the end, we can say that it's not a chance finding. So it's an assumption that feed into how you analyze the data. It's a critical assumption that ensure the validity of your analysis for you to be able to draw a causal inference from the analysis. I alluded already to stratification. The importance of stratification is to ensure that even within specific profile of patients, you have reasonable balance. Let's take, for example, specific characteristics like gender or region. Sometimes there's ethnic sensitivity where a particular drug or even the disease manifests differently in specific subpopulation. Let's say between male and female, or between much more severe categories of the disease versus mild category. If that is known, we don't want to leave it to chance. And at the end of the day, you end up with a split that there's an imbalance where one group represents one subpopulation, say male versus female, and thereby you have confounding effect of that particular characteristics with the effect of the drug. So stratification ensures that within each strata, you then randomize. So you have a reasonable balance. Your randomization reflects the targeted randomization ratio in specific strata. That's the importance of stratification. It's much more important in small studies, because in big studies where you have a large number of patients participating, usually randomization is expected to help you balance things out. But when you are in the context of small studies, it's very important to ensure that the factors that are prognostic for the disease, the critical one, are stratified for, they are controlled for as part of the randomization process. And when you stratify the randomization, it's also more efficient. It's often critical to stratify the analysis that follows as well. So the stratification factor will be part of your statistical analysis model. I move on to blinding. Blinding, again, is another principle that ensures that we do not unknowingly introduce bias into the conduct of the study, into how data is captured, into how patients are handled in the course of the study. So often the case in a blinded study, patients will not know what they are receiving. Why is that important? It's important because if they know they are receiving active treatment, there may be bias. They may have a predisposition that, okay, since I'm being treated with the active substance, this is how I'm supposed to respond. Yeah. It's also important also that physicians are also blinded. The physicians, the investigators interacting with the patient. And when those two aspects are in place, we often label the study double blind. But that's not where it stops. Even for the sponsors that is conducting the study, the sponsor staff are usually blinded as well, so that they don't know which patient is receiving what. And that is very important to keep the integrity of the data capturing process, the execution phase of the study, so that we don't query some data points more than we query the others, because we know what patients are receiving, and we have a predisposition as to how the response is supposed to be on one treatment or the other. So that's the importance of masking. And randomization is, it goes along with that, for you to have an effective blinding, it has to go with randomization. And we also, often the case, do it such that the packaging of the drug versus placebo or the comparator, they look the same. The drug, if possible, they will smell the same, they will taste the same, so that when patients see it, they can identify that, okay, I'm receiving placebo or I'm receiving the active. And all these are in place just so to ensure that we do not unknowingly introduce bias. In the interest of time, I'm going to move quickly on this latter piece, the content of the clinical trial protocol. There are multiple aspects that I highlighted already, but for us, session 10 is usually the data analysis session. This is where you specify, in most cases, how you're going to analyze the data that is being captured to address the objective of the study. You pre-specify different aspects of the analysis. The the endpoints. You designate which one is of primary importance, which ones are of secondary interest. You pre-specify the hypothesis. What's your primary hypothesis? How are you going to address multiplicity? We'll speak more about that. How did you determine sample size? The statistical model. How are you going to address missing data issue? Those are aspects that needs to be pre-specified as part of your protocol. These are just some references that we can look on later on, but in the interest of time, we will move on. I'm going to pass it on now to Brian Smith, who is going to start us up on the inferential testing part. Brian, do you prefer to show your slide, or I can move it for you? You're on mute.
Yes, sir. Okay, thank you. Hello, Dr. Edison?
Yes, I can hear you.
Oh, good. Thank you very much for this beautiful presentation. Though by the schedule, we're supposed to have entertain some feedback from our audience, from our participants, so that the people, I don't know whether that is okay by you, or you want Dr. Smith to move on with what? But I would have loved that we entertain one or two questions for you to remarks and why Dr. Smith is warming up on what you have just presented.
Let's do that then. Yes.
Let me give you this information again. It is very important that this presentation is being streamed live on YouTube. So if you are not with us within this environment, or you are not with us, you can join to via the YouTube live stream. And the president of the society is around here. We'll do the proper introduction later. So for questioning, if you have one or two things to ask Dr. Dini on the two papers presented, I think this is the time to do that so that you can rest that one before we call Dr. Smith to have some questions. Dr. Dini, you have a comment, you have a query from one of our elder professors here. Is council is a member of the board of trustees for us to go. Please go.
Thank you very much, Paul. I enjoyed your presentation. I would like you to comment on the level of theoretical development in the area of Bayesian decision theory involved in drug development. For example, set distributions, prior distributions, and procedural distributions that you will involve in taking decisions and drug development. Thank you.
There are questions.
So there are, Dr. Andy, can you take note of these questions one by one so that we can answer them together? And that will be fine by you?
Yes. Go ahead.
And there are some online questions. People have already posted some questions for you online, which you are going to mention to you shortly. So just take note. I'll manage to anyway. Okay. Thank you, Doc, for the presentation. I have some few questions. The first one is, you mentioned, you talked about recruitment. I'd like to know if the process of that recruitment follows what we know in survival analysis, where we have issues of loss to follow up, we have issues of patient being able or subject being able to join even when the study has started. Then how do you manage decline from patients who has at the start of the study agreed or consented to be part of the study and later decided not to be part of it anymore? Then the second question is is on NTD. Now, do we have a strong difference between NTD and ED? Okay. I know in bioassay we talk about effective dose. Today we talked about the maximum. We talked about the FTD. So I want to know a bit about that. Then the third question is, what do we do when we observe heterogeneous deviation from linearity? Okay, because in the process of presentation, I think the aspect of relative potency has not been discussed. Where do we have this? Where does it come in? The aspect of relative potency, where does it come in these drug development stages that you have mentioned? And how do we liken that to heterogeneous deviation from linearity, which seems to be a problem in bioassay? Thank you. Thank you very much. To allow effective response from the totality, can we please have your response to those before we take another set of questions? Online. Yeah. Over to you. Totality.
Oh, okay. Okay. Thank you. So I can quickly address the questions raised. So the first one being on the use of Bayesian statistics for development in Bayesian methodology. Yes, Bayesian methodology is well developed and in use in pharmaceutical development. More so in the early phases of clinical development, phase 1 and phase 2. It's used in those phases more than it is in phase 3. In those two early phases, in phase 1 and phase 2, it has to do a lot with internal decision making, right? Where it lends itself to, those phases lend themselves more readily to Bayesian framework, where the sponsor can introduce the notion of what is the prior belief about different aspects of the drug into the design. The challenge is in phase 3 development, where regulators are often not very too predisposed to the use of Bayesian methodology from the standpoint that there could be a difference in prior belief between multiple sponsors, difference between the regulator and the sponsor. So frequencies framework is commonly used in phase 3. But that's not to say that Bayesian method has not been used before. I'm just talking about the frequency of use. And in terms of how Bayesian methodology is used, again, a lot of work has been done in how it is used to make decisions, especially during the earlier phases of the study, even in how we manage portfolio, which compound is likely to have a higher probability of success than others. And so these are very important in terms of how decision is made. And I want you to also stay tuned. There will be a speaker on Wednesday that will talk about decision making in drug development. So aspect of this might be addressed there as well. But just as a general comment, that's what I would say about the use of Bayesian statistics. The second question was about survivor analysis. Of course, in this case, I didn't touch on the different type of endpoint that we can encounter, right? So the endpoint could be binary, it could be continuous, it could be time to event type endpoint, where survivor analysis that you talk about will be relevant. You also touch on the implication of a patient consenting and later withdrawing from the study. If they haven't been randomized into the study, that's okay. In fact, as part of the protocol, as part of the process, not everyone that consented are often randomized. They might consent to participate, but they are not meeting the eligibility criteria. So there are often screen failure, right? And then some patients will make it into the randomization. But maybe your question could also be, even after randomization, what if a patient decides to withdraw? And that's an experience that is common in practice as well, that you have discontinuation post-randomization. And those are very important issues that has to be addressed in the context of the study. What does the treatment effect mean, given the reality of this discontinuation that happened after discontinuation? What are we going to assume about this patient? Are there, and depending on the question that we want to address, different question might call for different assumptions about the patients that discontinue, about how we are going to impute the data for them, for the conduct of the analysis. Your question also touched on the difference between effective dose versus maximum tolerable dose that I spoke about. When maximum tolerable dose is established, usually when you move into phase 2 development, what it means is that you focus on the range between your no dose to maximum tolerable dose. So the maximum tolerable dose establishes a cap, a safety cap for you. And within that range of no dose to maximum tolerable dose, you are hoping that you will be able to establish a dose as being effective. You can have a minimum effective dose defined in that range. I think the last question touched on deviation from linearity, relative potency. And if you're still here, in the latter part of the workshop, my colleagues will speak on clinical pharmacology, so they might touch on the issue of relative potency. The only thing I would say in response to your question is that we do, especially in phase 3, there's an obligation to pre-specify how we are going to analyze the data that is being captured in the study. So how are you going to know which assumption is in place and which is not? And we do our best to learn from the different studies that have been conducted up to that point, and or even studies that have been conducted maybe in the same disease with other compounds, to learn from those studies to determine the assumption that makes sense for the current study. I will stop there, and maybe Brian, you have anything? I see you are on camera already. So if you have anything to add to what has been said, please go.
I was just going to add to the Bayesian component to it. If you think about Bayesian statistics to a certain extent, it impacts, one of the big parts it impacts is the prior. and one is the interpretation of the data after the fact, right? But as far as prior is concerned, we're more likely in early development to want to use a prior. One of the things that we could do, for instance, is sometimes we have placebo information, and so we can use historical information as part of the prior for placebo in order to augment our control group, if that makes any sense or not. And, you know, for decision making, a Bayesian framework is sometimes more natural to think about in the probabilities and Bayesian statistics. Always keep in mind, right, if you start with a non-informative prior, to a certain extent, I found at least that frequentist and Bayesian techniques tend to lead to the same decision. So therefore, you know, sometimes they're exchangeable, if that makes any sense whatsoever. The other place for Bayesian statistics is sometimes the problem itself is hard to solve without MC methods. And so therefore it becomes more natural to use a Bayesian framework to set up the problem. But if that sort of kind of gives you a perspective, but regulators in essence, and Neat pointed this out, tend to want to rely on the data and not any prior, if that makes any sense.
Okay, thank you very much, bro. We have some participants online that want to ask one or two questions, and we want to listen to few of them. We're not able to accommodate all of you, but because of time, what we are by the schedule, we're supposed to do for lunch right now, and listen to Dr. Brand for the presentation. So, Dr. Banjo, are you ready for the question?
Yes, sir. I'm ready with my question, sir.
Very brief, please. Very brief.
Okay, thank you, sir. Thank you, Dr. Adewale, for your presentation. My question is, when you were discussing the phase 2B, those finding stage, you talk about obtaining the optimal dose, which is a trade-off between the efficacy and safety. So I want to ask, sir, when you are looking at the efficacy, the trade-off, do you consider the patient demographic factors such as age, gender, region, and so on and so forth? Do you consider that? That would be my first question, sir. My second question is that you talk about the statistical analysis plan, the SAP, which is usually before the clinical trials. Now, before the data is being even obtained, does this change? Because sometimes the nature of the data that we have might want to give us a guideline on what type of analysis to use. After the you would have the plan, does it change at the end of the day when you have already generated those data? And then lastly, sir, you said patients are not privy to know the type of active or the type of drug they are using, maybe it's a placebo or not. I want to ask, sir, will this not violate the patient's rights? Thank you, sir. These are my 3 questions.
Thank you.
Okay.
We want to come on the second question. Dr. Adoni, please take note of that. Anthony Ebo, are you there? Anthony?
Yes, I am here. Thank you so much.
Very brief, please.
Thank you. Okay, it's going to be brief. Thanks for the opportunity, sir. And thanks too to Doc for the very wonderful presentation. Yeah, my question is concerning when you mentioned something pertaining to the therapeutic window. You didn't say whether it gives a particular confidence level where the number of patients in the sample size falls within before you accept that. So, because I was thinking that maybe you will have specified that the therapeutic window will have been within a particular confidence level for the patients who you really do consider. Thank you.
Okay. Should I go ahead, Prof, or you want to take more? Okay. We can't hear you.
You can't hear me?
Okay, now I can hear you.
Oh, sorry. I think you listen to the two persons that asked questions. Okay.
I can move on and address those.
Address those two. So that,
okay, good. So the first question is about those findings, about the effect of covariates. So do you use the example of age or gender? And there are, yes, methodologies that allows you to incorporate the covariates in the analysis as part of your those finding analysis. I'm not sure if you might be familiar with a particular method called MCP mod, multiple comparison procedure and modeling. It's a framework that is very efficient to address what you just talked about, where on one hand you are modeling the dose response relationship, at the same time doing dose comparison, comparing dose to placebo, trying to establish a significant dose response profile, and establishing which one say meets your clinically relevant threshold of effect. So yes, that can be done, is the answer to that question. Beyond that as well, at the end of the study, you can also do descriptive summary by different subgroup factors, just to better characterize what you are learning from the data. The second question touched on pre-specification of the analysis. What do you do if the data that you capture at the end doesn't align with what you have pre-specified? So the way I will address that, very often you know the nature of the endpoint that you are going to capture. If it's a binary endpoint, that won't be a surprise to you. You already know because you designed the study. If it's a continuous endpoint, you know. So that level of information you have going into the study. Where you can have potential misspecification issues. Let's say that you are planning to analyze a continuous endpoint using linear model, which we all know we require certain assumptions for validity. And at the end, let's take, for example, that you don't think your data is behaving normally. Not the normal distribution assumption is being violated. Hopefully, that is not happening on you in the context of a confirmatory study. Yeah, because at that point you've learned a lot from previous studies to be able to come up with a reasonable analysis that that will satisfy the relevant assumptions. But if you happen to be in a situation where there are anomalies that you did not anticipate, yes, you can do additional sensitivity analysis, that is more appropriate for the data that has been captured. However, credibility is an important issue in drug development. It's not just your interpretation of the data, it's also the interpretation of the regulatory authorities. So you want to make sure that you are doing things in such a way that the statistician at the other end of the table will agree with what you have done, how you have approached the analysis, that you are not just changing the method to suit your own objective, what you hope to see, what you hope the data tells you. So that's why pre-specification is important. And in fact, it's often the case that we don't want to do any amendment if possible. Have post database lock. It will have to be a very extreme situation before you can go into that kind of post database lock amendment of the statistical analysis plan. The last question also touched on those response where you are talking about the confidence level of the therapeutic window. And I will briefly answer that. Yes, when you define the therapeutic window, it's a range, but also even the minimum effective dose, which is defined by they usually defined based on efficacy. Yes, you put uncertainty around that, which we like to express by confidence bound. Yeah, so that can be done. So it's a therapeutic window, but when you put in the limit around the boundary of each limit, you account for the uncertainty there. Okay, I will leave it there in the interest of time. Prof, if it's okay with you, Brian can take it off.
There are a series of questions of our participants online that have been sent to you. We want you to address to that one shortly. But generally, I want to ask this question. What Novartis does over here is the research in drug development. And in your intro, on drug development, you give credence to the usage of herbs, international herbs all in Africa. And meaning that some of the herbs that we are using Africa to ask of the level of efficacy at least one or two things. Only maybe the combination of doses to be used. How much of these local herbs have you been able to investigate for their potency novelties over the years? And what has been the success story from your hand? Do you have any response to that?
Yeah, thank you for that question. I have to say that it's I really don't can't tell you much about investigation of local herb. The area of my work is in the latter part of the development spectrum. My colleague Brian is closer to the early development space where discovery happens. But it's often the case that here we have a library of compounds that has been studied, and there are known the molecular profile, the characteristics of the compound is well known, and scientists in the lab sometimes based on what they are targeting may want to synthesize a compound that have specific characteristics. I'm not aware that there's something that comes from a local environment in terms of herb that is going into, say, a pharmaceutical laboratory to investigate. That's not what, I'm not aware of that. Maybe Brian can comment more. You're on mute, Brian.
On this note,
we have lots of compounds. We don't even know what they're for. We have catalogs. We buy them. Some of them come from natural products. We have lots of compounds, right? We do that initial screen that Nee just talked about, and we come up with hits. That is, compounds that have activity in whatever that we're looking at. Then the interesting thing happens. Analytical chemists then take those hits and they start changing the molecule to try to get properties that they want from the molecule. So usually what's ever in our hits sort of thing isn't end up being the final drug. It's like a scaffold where they do other things to the molecule to try to make it even better. And so they spend a lot of time doing that, running experiments of trying to get a better molecule. So I don't know if that helps from your perspective, but that host of compounds that we start with could be natural compounds that you find in nature, or it could be something that we purchased from a university that they put together. So I don't know if that helps at all.
On this note, I want to enjoy Dr. Adeni to take note of some of the comments by our online participants. By the time Dr. Bransmith is through with his presentation, we have the opportunity to respond to a few other questions raised by our online participants. On this note, I want to call on Dr. Brand to comment his presentation. Thank you very much.
I think Brian is setting up to share his screen.
Okay. Okay.
Please take your screen down, please, Dr. Adeni.
Okay.
Okay, so now, can you guys hear me okay? I'm going to share my screen.
Please go ahead. You can share it.
Okay, and then I'm going to open my part of the presentation. You should be able to see this now.
Yes, we can. Please go ahead.
Okay. Very good. And one of the things I did is I just put in earphones because I was having troubles here in the conference room, but I hear it a lot better now. So I hope so I hope I can hear your questions better. Okay, so what I'm going to do now is introduce sample size estimation. And this is one of the key component of a clinical statistician's job. And let me talk to you why it's so important to get this right. There's an ethical reason, right? Until the drug's approved, there's questions about the drug's efficacy and safety, right? And so if we have too many subjects in a trial, that means we're exposing patients, more patients than what we need, to something that either won't help them or potentially won't help them, or to something that could cause them harm. Right? So we need just the right amount. And too few subjects means we can't determine if the compound's effective or intolerable. And if we took that to an extreme, that would mean no new medicines, right? For us in the pharmaceutical industry, there's also a cost reason, right? Too many subjects means you're wasting money by acquiring more information than needed. Too few subjects means that you don't have a chance to figure out the thing that you want to figure out. Right? So these are the reasons why it's so important to try to get the sample size right. Traditionally, we talk about this in terms of type 1 and type 2 error rates and power, and I am going to talk about that, but I'm going to try to generalize it to a certain extent as well, right? But just to try to keep this simple, suppose that I have two treatments, our treatment and placebo, both with a mean, both with a true mean, right? Our one-sided hypothesis is that the mean difference is less than or equal to 0, versus we have a treatment effect that is that the mean of treatment is better than the mean of placebo, right? We use observed sample data to test the null hypothesis, and any hypothesis test is subject to incorrect decisions. And there are two types that we tend to call type 1 error. Type 1 error is when we reject the null hypothesis when actually the null hypothesis is true. That is, we're saying something is working when it's really not. That's a regulatory risk. There's also type 2 error. That's when we fail to reject H0 when HA is true, right? That is when we're not able to show that something that's good is good. That's the drug developer's risk. All right, so what we're interested in is error rate control. We pre-specify the significance level, the type 1 error rate, i.e. alpha, right? Oftentimes we set that equal to 0.05 for a two-sided test that has been a tradition in statistics for 50 years. We can argue about whether or not that makes sense or not, and I would argue it doesn't, but we need a value. So alpha equal 0.05 two-sided, or alpha equal 0.025 one-sided. And I think Nee's already used that number before. We then also pre-specify the maximum type 2 error rate, or beta. For instance, we could specify 10%, right? The power is 1 minus beta, so therefore the power would be 90%. 90% is used a lot of times in confirmatory sorts of trials. In the trials I work on, which is in early development, oftentimes we use 80% power, right? But regardless of all this, we have a decision table that kind of looks like the following, right? We have type 1 errors and type 2 errors, depending upon whether or not the null hypothesis is true or the null hypothesis is false. Now, of course, we don't know whether or not the null hypothesis is true or false, or otherwise we wouldn't be doing the study, right? But these are the errors that we potentially fall into. All right, so type 1 and type 2 error rates and power, right? The sample size calculation requires all of these, and it's an exploration of the relationships between the type 1 error, the power, the treatment difference, how big of a difference is that alternative hypothesis, how big are we wanting to see, the variability, e.g. the standard deviation, and the sample size, right? So we're trying to take all of those things into consideration. Now I'm going to generalize this motive, and let me give you the motivation. Not all drug development questions are formulated this way. There are parameters other than means, right? Hypotheses that are different from mu2 minus mu1 is less than or equal to 0, right? It's not explicitly stated here, but this language largely implies frequentist statistics as opposed to Bayesian statistics, right? So I want to generalize in such a way that the thought process is the same for these various differences, that it kind of all fits into one umbrella. It's actually relatively easy to do, right? Through replacing how we're thinking about these things, right? So type 1 and type 2 error formulation, we think of alpha, the type 1 error. What I'm going to change that to in our thinking is success criteria. I need some sort of criteria in which I will say after the end of the trial that the compound's been successful, whatever that means exactly. 1 minus beta, which is the power, right, is the probability of success given some truth or assumed distribution, right? So it's the probability of success. It's how likely that you're going to pass that success criteria, right? The treatment difference and the variability. All these things are actually about your assumptions about what the assumed distributions of treatment 2 and treatment 1 is, right? So if I have 2 distributions that I'm assuming, I can figure out what the power is right? What the probability of success is of saying that those two things are different through my success criteria. The sample size is the same sort of notion. So I hope that that kind of helps clarify that it's more than just type 1 and type 2 error, although type 1 and type 2 error is an important thing to understand in and of itself. And probably, you know, since many of you are statisticians, you already know all this anyways. So what do we need to get this right? Well, what we need is a well thought out question of interest. You'd be surprised how often the clinical team doesn't really have, at the start of a study, a real precise question that they want to answer. We also need a well thought out endpoint that could answer that question, right? We need a decision rule that indicates success of the molecule for the trial. We need the assumed distributions of the endpoint for the molecule and placebo group, so that if true, an assumed mean difference from placebo will be just barely clinically meaningful. And I note it doesn't really have to be means, it could be something else. We need the chance of success given the assumed distribution, that is power. All right, we need a well thought out question of interest and endpoint. These are actually clinical scientists' responsibility and often obvious, but sometimes it's not obvious. This is especially true where I work, which is in early development, and a disease has not been frequently studied. The statistician's role, when it's not obvious, is by understanding literature data, you can help the clinician understand quantitative limitations of some questions and understand the quantitative properties of different endpoints. Okay, so that's kind of what we would do in that to help with that. We need a decision rule that indicates success of the molecule for the trial. The typical decision rule, as I've already said, is a two-sided p-value less than 5%. This is absolutely true in phase 3. And in fact, you have to interpret, and I'm going by the Food and Drug Administration in the United States in this, they basically require two separate phase 3 studies that are confirmatory at the 5% two-sided level when comparing to placebo. For other phases of development, larger type 1 errors are often warranted. And we certainly use other than the one-sided 0.025 for that. In fact, the decision rule does not have to be a comparison to placebo. There's a whole set of issues and something called non-inferiority analysis that are done. There are other things, other sorts of hypothesis. We'll be talking about equivalence later in when we're talking about clinical pharmacology. Sometimes it can be just an observed clinically significant value, right? This can happen relatively early on. So no p-value at all associated with it. Or sometimes we can use a Bayesian framework, as I talked about. The statistician's role is to help the team negotiate all these possibilities, right? To try to get everything clear. We need all that clear so that I can actually get to the sample size. Okay, we need the assumed distribution. Of course, these are things like, is it a normal distribution, a log normal distribution, a binomial distribution, et cetera. Also, the assumed difference between treatments would be part of this. These are assumptions that we would be making. If you're, say, using the normal distribution, you have to assume what the standard deviation is, right? So, you know, things that you're doing at this point, you know, hopefully you have either literature or actual data where you can get a strong idea about what you think the standard deviation is, for instance, right? And you can have discussions about, you know, what do other drugs on the market, for instance, what sort of difference do they show? Do we need to show that difference? Do we need to show more than that difference? If that makes any sense. The smallest clinically meaningful difference is the clinician's responsibility, although we can help providing information. looking at literature and in-house data, the statistician role is to estimate the standard deviation well. Okay. We need the chance of success given the assumed distribution. That's the power. How have a chance as a team wish to succeed if the truth of a clinically meaningful difference is really true, right? And that's dependent. And as I'll kind of say this later in the last part, but just keep in mind, right? Just as far as cost is concerned and complexity, right? We're talking about in early development, $5–10 million to do a study. We're talking about in phase 3, $100 million, $200 million to do a study, right? Obviously, the risk, if the power is the sponsor's risk to a certain extent, you would change that. You want a lot of assurity when you do phase 3 that your power is really high. Early in drug development, you don't need as much or as high of a chance. Okay, so there are 2 ways of doing a sample size sort of calculation. Sometimes the mathematics is such that there can be an analytical solution. If the mathematics is too complicated or intractable, one can examine sample size through simulation. I'm going to show you an example with both. Not to overwhelm you, but the same sort of ideas go into any sample size sort of calculation that you would do. So example 1 is an analytical example. What I have here is a situation where I'm going to compare two proportions. The example is drawn from a study in something called chronic spontaneous urticaria, and urticaria is basically hives, you know, red things popping up on your body, and these poor individuals have these itchy hives happen to them relatively regularly, right? It's not pleasant in any sense whatsoever. I don't think it's life-threatening, but as long as you have those, you can also have headaches and other things. I mean, your quality of life is pretty low, right? There's something called the weekly urticaria activity score, right? It's basically just a patient diary where people are counting incidents of having these hives pop up during a week, right? And so therefore, it's thought that having less of these things in a week is a good thing. And so we're going to compare placebo and drug at week 12. That is 12 weeks after being treated with the drug, right? Say on a daily basis. We're going to compare the proportion of individuals that are less than 6. So let's just assume, not just assume, but the placebo and drug now then have Bernoulli distributions, right? With a probability of success for placebo of pi1, a probability of success or less than equal to 6, pi2. Let's see. And we're interested in the one-sided null hypothesis that pi2 minus pi1 is less than or equal to 0. Previous trials suggest the response rate at week 12 is of about 12% for placebo, and we hope it's 40% for drug. That's when we think it's a meaningful compound for us. So a way of comparing the 2 proportions, again, we know that if we have a binomial distribution, asymptotically, we have normality starts to apply, right? And we can consider the z-score, which is the difference between the estimated proportions on both treatments divided by the standard deviation times square root of 2 divided by n. In this particular case, because we're dealing with proportions, and if you remember all this, the standard deviation sigma hat is actually estimated itself from the pi hats themselves, and there's a formula for it. This is asymptotically normal 0, 1 when delta is equal to 0. That is when there's no treatment effect. Okay. Okay, so we want to choose a success criteria such that the type 1 error weight is less than or equal to alpha. So we reject H naught if Z is greater than Z1 minus alpha. And based upon this, we can come up with the power function. That's the probability that Z is greater than Z1 minus alpha, that is that we reject, given that delta, the difference, is greater than 0, right? And that probability is written down there. It's the probability that pi 2 minus pi 1 minus delta over the standard error is greater than Z1 minus alpha minus delta over sigma over square root of 2n, right? Given that delta is greater than 0, right? And so we have this formula, right? It basically has a normal distribution, or 1 minus the probability coming from normal. The power depends only on the difference, pi 2 minus pi 1, but also on the individual parameters, pi 2 and pi 1, parameters via sigma squared. For a fixed delta equal delta A, we can solve this question, and we obtain the sample size formulation that n is greater than or equal to 2 times the z-score of 1 minus alpha plus 1 minus beta, which is the power, right, squared, or 1 divided by delta A over sigma squared. And with that, we can just crank through these are different observations of pi 1, right? Different observations of pi 1, different deltas, and these are the sample size given all these different combinations based upon that formula. Right, so now we've investigated what different things can do. We can go back to our original problem. If we want alpha equal to 0.025, we want beta equal to 0.1, we want pi 1 equal to 0.12, and we want pi 2 equal to 0.4, right? We can actually plug all those numbers in and come up with the sample size, which is 48 subjects per group, right? Not particularly too difficult, right, when you have an analytical solution. But notice there's a lot of assumptions that I have to make in order to do that. And that I didn't talk about how much work it is to try to make sure that those assumptions are right. That's the key to it. The actual mechanics of calculating the sample size after you have the assumptions, easy, right? Getting the information so that you think it's relatively good, maybe not so easy. Okay, example 2 is I'm going to show a simulation example. I'm going to use the same exact setup, right? So as not to throw anything for a loop. It's the same situation. Y1 is Bernoulli pi 1, Y2 is Bernoulli pi 2. I'm still interested in the 12% and 40% number. Instead of relying on the central limit theorem, however, I'm going to use Fisher's exact test. Now, I believe that there are many approximate analytical solutions to the Fisher's exact test problem for figuring out the sample size. But just to show you simulation, I'm going to ignore those. I hope you don't mind, and go right to showing you how to do it through simulation. Now, Fisher showed that the conditional on the margins of the table A is distributed as a hypergeometric distribution with n draws from a population with A plus B successes and 2n minus A minus B failures. It's actually more general than that. Notice that I have this written in such a way that the sample size on treatment and the sample size on placebo are the same, n and n, right? This could be done observationally, and you could just put A, B, C, and D for those top 4 cells and not worry about the n's if you didn't know what the n's were. But anyways, regardless of that, that's where we're at there, right? I'm going to just show you this again. The probability, so have A and B, right? That is the number of observations on treatment that are less than 6, the number of observations on placebo that are less than 6, I can calculate what that probability is, right? It comes from hypergeometric distribution, and it's, you know, A plus B, choose A, 2n minus A minus B, choose n minus A, divided by 2n, choose n, right? And you can figure out that probability. It turns out that if you want to find the p-value, right, the p-value would be what the p-value is the probability that you got what you got or something more extreme, that or something more positive than what you got, right? It would be just the sum from the hypergeometric distribution from I equal to 1 to B, right? Basically, if you want to think about it to a certain extent, right, a better situation would have been getting, you would have had a larger treatment difference if you showed no one on placebo had less than or equal to 6, right? Or 1 is less than or equal to 6, all the way up to B is less than or equal to 6. So if you sum all these things together, you have the p-value. All right, so how do we simulate? Now that I know how to figure out what the p-value is, right? We pick a sample size, say n equals to 50. Assume, for instance, that pi 2 is equal to 0.4 and pi 1 is equal to 0.12. Randomly generate 50 binomial observations for pi equal 2. I should have written Bernoulli there. Sorry about that. We randomly generate 50 Bernoulli observations for pi 2 and 50 observations for pi 1. Calculate the one-sided p-value and note if it is less than 0.025. All right, that's your success criteria. Repeat, say, 1,000 or 10,000 times. If the proportion of success is less than 0.9, repeat with a larger sample size. If the proportion of success is greater than 0.9, repeat with a smaller sample size. Continue to converge on the appropriate sample size. I did this for this particular problem, right? Here I run 10,000 simulations for each one of these sample sizes from 50 to 60, right? And you see that around 55 subjects is when the probability or the power is about 90%. So given the simulation, I would go with a sample size of 55 per group, if that makes any sense. So what are the conclusions from all this? One of our most important jobs is to get this right. Thinking of type 1 and type 2 error is useful. It can be generalized to other questions. The calculations often can be analytical, but sometimes your resort will be to be able to do this through simulation. And with that said, I know this wasn't that complicated, but I'll be happy to stop and take any questions that you might have. Let's see here. I'm gonna stop sharing so that.
Thank you very much.
Any questions?
Yeah. Thank you very much, Dr. Grant, for this beautiful presentation. Want to take a few questions before we go to the next lecture.
So I'm not hearing anything, which that's okay. And if there's no questions, it was rather simple, and I get that. We can get to the next section for me if we'd like. I believe he's got the next component.
Okay.
Sorry, there are some questions online. Dr.
Okay.
Adewale said you should attend to them. There are some questions.
We have some questions already that we were able to take where Dr. Adewale was, after Dr. Adewale's presentation, we are going to take some of these questions, and while we are waiting for questions on the presentation of Dr. Graham Smith, our participant. Dr. Adewale, are you there?
No, he stepped out for
Okay.
for 10 minutes. That is
okay.
I'm posting some now.
Well, let me open the floor by this question, Dr. Smith. In your presentation, sir, are you hearing me? Dr. Smith, are you hearing me?
So I'm not. I mean, I hear people are speaking, but I'm not hearing. Let me try taking this out. I'm just not hearing it very well.
Okay.
To be honest with you.
Yeah, you let us know that in your presentation that the difference of under the test of the proportion, when you are comparing two proportions, the pi 1 and pi 2, each of which follow binomial distribution. That is the underlying random variable that follows the binomial distribution, given the power and factor of the parameters. You let us know that the delta, which is the difference between the pi 2 minus pi 1, is not only the one that determines the size of the power of the test, but the size of each of these proportions itself. Taking, for example, pi 1 as the threshold, maybe the standard or the placebo, and the pi 2 want to investigate the proportion. How does it differ from the pi 1? In your simulation study, what desirable size of power are you suggesting? Given the size of this pi 2, especially relative to pi 1. I get that.
I'm not sure I heard it all, but in general, for proportion, right? Unfortunately, because the variance of the difference is dependent upon pi 1 and pi 2, you can't just know the difference to figure out what you can't just have a difference that you're interested in to figure out what the sample size is.
Okay.
Basically, so it's dependent upon, what you're assuming about what the actual pies are. I hope that leads the. And Nee's asking to be unmuted, so if you would do that, he might be able to. He may have heard better. I'm not sure. So if you can allow him to be unmuted.
Okay.
Okay. Yeah, I think I'm unmuted now. Yeah.
Okay.
So I had the question. So when it comes to difference of proportion, yes, the magnitude of the difference is important, but also where you have let's say the reference, the control proportion on the probability scale is important, which determines your variability. Yeah, if you are close to the middle of the probability scale with the reference proportion, whether the reference is pi 1 or pi 2, that's the highest variance possible. And therefore, you have increased sample size. Usually, at the either extreme of the probability scale, you only need a small difference to be able to have decent power. So depending on where you are on the probability scale, that has effect as well, in addition to the magnitude of the treatment difference.
Right. Right. And I mean, to a certain extent, naturally speaking, right, difference in proportions, I mean, it's sometimes natural to think of them as, say, odds ratios or just relative risk between them, right? Which, you know, you can transfer one into the other, which changes the problem to a certain extent.
Okay. Okay. And then again, Dr. Smith, in your simulation, when you demonstrated you to us under the simulation, that is determining the sample size via simulation, and we discovered that in the determination of the power, none of your power is just in the neighborhood of 90%. Are you comfortable with that level of power, or is there a proposal from your end that to increase the power, of course, by increasing the sample size? Because when you
So am I satisfied with 90% power in general? I think in general speaking, there's two components here. Hopefully you're talking about the minimal difference that you're interested in. So we don't actually know, to a certain extent, our drug could actually be better than that minimal difference, right? So if that's true, then I have an even larger chance than my power of being able to detect that there's a difference, if that makes any sense. I think when you start getting into powers larger than 90%, though, you start just getting into feasibility issues associated with these studies can already be large. I mean, it's not unusual, you know, in a survival analysis case. I mean, these were relatively small, but you can be in situations where your sample size is 5,000 per group at that point, right? If you're doubling that, because you're going up to more power. So if I go from 90% power to 95% power, that's a pretty big jump at that point. So if I'm doubling that sample size, that's a lot. So I think that that's part of it too. So typically 90% is usually what we're seen as the largest sort of power that we would take into a clinical trial.
Okay. Thank you very much, Dr. Smith. We have some online participants that have one or two questions for you. Dr. Ali Usman, are you there? Dr. Ali Usman, are you there?
Why is there? Can I ask a question?
Yeah.
Okay. Doc, well done. I hope you can hear me.
Okay.
Yes, my question is, when we're talking about assay that is conditioned on leader dose, okay, where we are interested in dead or alive, would you still advise that we go ahead with your test of proportion?
No, not necessarily. I mean, I wasn't recommending a test of proportion. I was just showing it as an example. With proportions, you get into all kinds of different possibilities that you can do. And you said dead and alive to a certain extent, which brings in the possibility of how long that it took to get there is a possibility. But no, I mean, I would need to know more about the problem to know what analytical technique I would be happy to use with it.
That's fine. But now, at what point is the power of the test important in the determination of sample size?
At what point is it determined? Well, I mean, I think in general, we can just go through. I kind of think at each stage of drug development, right? In essence, we're trying to get the right amount of information at the particular time for the particular question. The power helps, along with what the test is, helps determine what that right amount of information is, right? So I think it's important. I think it's the only way that you can get to the sort of notion of how much information do I need in a particular trial. Is that helpful?
Just something to add to that quickly, Brian. if you think about it in terms of the converse of the error, right? The type 2 error. So we do our best to make the right decision. And so if you think about it in terms of not making a type 2 error, a false negative. So it's always important. If you think about it from that perspective, it's always important throughout the drug development spectrum. It's just that as you move along, the power consideration, there's an ethical side of it that Brian already alluded to, that you have sufficient power that can answer the question of interest. And that's always important, whether you are in early phase or in phase 3. In the early phase, though, typically we will admit a little more actually on both sides of the error, we will admit a little more error than in the latter phase of development.
That's a fair statement. But I think the concept of power is universal, no matter what sort of study you're doing. If you're going to invest money into something, right, or you're going to have an ethical issue with having individuals in it, you want to try to get to the right number without the concept of power, that is probability of, without a success criteria and some sort of probability associated with you meeting that success criteria. How will you ever know that?
Thank you very much, Dr. Smith. We have one participant. I want to ask one or two questions. Dr.
Yeah, thank you, sir. Thanks for the presentation, Dr. Brian. So I want to ask, since you are talking about sample size calculation, so from the planning stage, how do we identify whether the sample size is going to be of a superiority trial or equivalence trial or non-inferiority trial before you consider the power and even the level of confidence to use?
All right, you're asking if I change the question from a hypothesis test to a non-inferiority question, how would I go about sizing that trial? Is that what you're asking? I assume that's what you're asking. I mean, it starts with the same sort of concept. So in a non-inferiority trial, the first thing that you would have to set up to a certain extent would be some sort of level, some sort of clinical level, which you thought was equivalent or wasn't clinically meaningfully different from the active arm. And then basically what you're doing then at that point is trying to show that your treatment, right, your treatment is at least as good as that clinically, that clinical difference, right? So once you've set that up as your decision rule associated with it, if there's an analytical solution, you can go for it, or you can do it through simulation. I mean, I think that the key is that your notions about what a success criteria, it depends on the situation, but usually I've seen people frame them as one-sided confidence intervals, right? You're successful at the lower half of my one-sided confidence interval is above whatever I think that clinically meaningful, not clinically meaningful, unclinically meaningful differences. And then power would be just what sort of risk that you wanted to take associated with it. So I don't know if that helps.
Thank you very much. Thank you very much, Dr. Smith. Thank you very much for teaching us today's very important concept of a sample size determination when we are going to consider inference and some associated issues. So I want to move to the next lecture. I don't know whether it's going to be taken by Dr. Brasmith again or it's going to be taken by Dr. Oliver Santa. That's the influential issues and differential statistics and also statistical issues as relates to multiple hypothesis tests. Who is taking us that? Smith?
Yeah, I will be taking that.
Oh, good.
And I'm sharing my screen. Let me know if you can see.
We can see you. You can go ahead.
Yeah, go ahead, please.
Okay, thank you. Yeah, so multiplicity. I think let me start by saying that the question of multiplicity apply to all clinical phases of development. However, it's much more important and much more emphasized in phase 3 part of the development. Yeah, and the reason being that we want to ensure that what is taken to the market, what is being administered in patients, there's sufficient evidence that it's not because we have a false positive finding. We want to establish beyond a reasonable doubt that this is actually providing benefit to patients. So why does this even matter? Let's start from there. And this is a setup. So let's say that you have an hypothesis test, whether it's a superiority test, whether it's a non-inferiority or equivalence test. The significance level is predefined at the 5% level, alpha equals 0.5. The question is, under that setup, what is the maximum type 1 error rate? 0.05. So if alpha is controlled for the test at 5%, your type 1 error rate is capped at 5%. That's what it means. But let's say you now have 2 independent hypotheses, each of them tested at 5% level of significance. And we ask the question, what is the probability of rejecting at least one of the 2 null hypotheses? Okay, so what is your type 1 error rate? In other words, in this case, the probability of rejecting or falsely rejecting an otherwise true null hypothesis. Yeah, what's the probability of rejecting at least one? It's simply 1 minus the probability of not rejecting either, right? So we can establish that the type 1 error under that case, even though each of the hypotheses is controlled at 5%, you can see that the type 1 error is almost getting to 10% under that simple scenario where you have 2 hypotheses. Yeah. One possible solution to this, that this is any topic or any discussion of multiplicity, you hear about Bonferroni correction, where you essentially have the allocated alpha. So for 5% two-sided, you are doing 2.5 for each of the 2 hypotheses that you have. And under that case, you can see that the overall type 1 error is controlled at the desired 5% level. Now you can take this further to the case where you even have 3, 4 or more hypotheses, right? So the probability of type 1 error will increase with the number of hypotheses that you have. Yeah, so in a case that you have 10 hypotheses, each of them being controlled at 0.05, the probability of type 1 error is about 0.4, 40%. Yeah, just because you are testing 10, even though you are controlling each of them at the desired 5%. So that's the issue with multiplicity. And each of these plots here represent the targeted alpha that you are controlling each hypothesis at. Yeah, and regardless of the alpha, you can say that there is certain number of hypotheses that is almost a given, that you're going to have false positive. Yeah, that's what is being illustrated here. Okay, I hope people can still hear me. So I just saw a message on my screen. So what are the sources of multiplicity in clinical trials? And multiplicity can arise from different aspects. It can be from the fact that you have multiple endpoints based on which you are trying to establish efficacy, right? So you can have multiple parameters that you are looking at. You can have different comparisons. In a case of the multiple sclerosis example that I brought on earlier on, where you have 2 doses in addition to placebo. Yeah, so in that case, you can compare each active to placebo. So you have 2 comparisons that are of interest under that setup, right? If you imagine having even more than 2 active doses, you see that the dimension of the multiplicity can grow very quickly. So you can also have multiplicity when you have multiplicity issue, when you have interest in multiple time points. Let's say that it's a study where you are following up for a period of 5 years. You assess the primary endpoint at year 1, at year 2, at year 3, and so on. And if you leave it open such that efficacy can be declared at any of these time points, that's a multiplicity issue because there are potentially more than one way to declare success under that setup. So you can also have it in a situation where you have interim analysis. We'll be talking on that later on as well. Or in a setup that you're looking at multiple populations in the same study. So those are different possible source of multiplicity or different combination of any of these. So as drug developers, as sponsors of the development program, we are always concerned that we don't want to keep investing in something that is a false positive, especially at the early phase of development. We want to be sure that we are not banking on a false positive signal, invest millions of dollars into development. Also, on the part of the regulators, they want to ensure that what they are proving to be used in patients, they are not based on a potentially false positive claim, that the drug is in fact going to do what we're claiming it will do in patients. Otherwise, you can imagine that you risk treating patients with a drug that is otherwise ineffective. So there are different ways to address multiplicity problems in drug development. How do we do it? These are some of the ways. You can reduce the number of questions that is being asked. In most cases, we try to streamline and to prioritize and say that this is the primary endpoint. This is the primary question of interest, and all other things might be secondary. Okay, so we'll define key questions that are going to serve as the basis of label claim. Otherwise, imagine if you have tens or hundreds of endpoints, as we've talked about in this simulation exercise that was presented. You see that it's almost a given that you will find a positive if you look at multiple dimensions of getting to a positive result. The other approach is to minimize the number of variables. What do I mean by this? You can have multiple endpoints, but you are defining a composite endpoint. In the case of repeated assessment that I used earlier on, you can create a composite endpoint. Maybe it's based on an average score over some specific time points. Or in the case of binary events, you can define a composite as the occurrence of any of multiple events. So in that case, the endpoint still translates to one particular event, as long as you have any of multiple events that you are looking at. So that's one strategy. I talked about prioritizing the question already, and also being clear in the protocol. What is of inferential interest? What are the questions that you want to do hypothesis testing on? And what are the endpoints that are exploratory in nature, that you are not going to draw definitive conclusion? So this level of clarity is what is important in dealing with multiplicity in drug development. And if you've done all this and you still have aspect of multiplicity, then a multiplicity adjustment strategy should be considered. And in fact, I would say that it's expected according to most regulatory guidance, especially when you are in those confirmatory type studies in phase 3 development. So now let's get into some mathematical notation in terms of how we will set up a specific multiplicity strategy that we're going to consider for the purpose of this presentation. So let's say that you have M different inferences based on M parameters, theta 1 through theta 2. And the individual null hypothesis is so defined. It's the hypothesis that is equal, each of those is equal to 0 simultaneously, as against the hypothesis that at least one of them is not equal to 0. In this, you can have it in a case where you are comparing multiple endpoints. Like I said earlier on, you are comparing drug to placebo on multiple endpoints. This can arise under that setup. Now, there's the notion of family-wise error rates. And a multiple testing procedure is set to control family-wise error rates at the level alpha if the probability of rejecting at least one null hypothesis is controlled at level alpha. Okay, essentially, we're saying that after incorporating the multiplicity adjustment procedure that you want to follow, we are back to the same thing as though you have just one hypothesis, right? You want to ensure that your type 1 error, regardless of the fact that you have multiple hypotheses, the type 1 error is controlled at level alpha. And there are different ways that you go about to do this, to adjust your p-values under the specific multiplicity procedure to attain this objective, to control family-wise error rate at the desired level alpha. In the most simplest case, for the Bonferroni method, what you do is simply to change. So if you have Hem hypothesis and your objective is to control overall type 1 error at level alpha, then if you simply control each of the hypotheses at alpha divided by Hem, that guarantees that you have overall control at level alpha. So there is the notion of adjusted p-value, which is in fact the smallest significant level at which a given hypothesis is significant under the multiple testing procedure that you are implementing. In the case of the type 1, in the case of the Bonferroni method, it's the adjusted p-value is the minimum of the multiplication of the number of hypotheses and the specific p-value, the minimum of that, and 1. This adjusted p-value using Bonferroni adjustment will result in overall type 1 error being controlled at level alpha. The method that we would like to discuss today is what is called the graphical testing procedure and the closed testing principle. In general, graphical testing procedure are approaches that can be visualized, and they are very useful in clinical development, because in clinical development, everything that we do is a collaborative effort where you are working with non-statisticians. So graphical approach lends themselves to ease of discussion with our non-statistician colleague. You can illustrate it and they can see how it flows. I would say that every graphical testing procedure corresponds to a closed testing principle, but the converse is not necessarily true, that it's not the case that every closed testing procedure can be expressed as a graphical testing procedure. What are closed testing procedure? What is the closed testing principle itself? This is a principle that is used in practice to construct powerful testing procedure based on the basic procedures such as Bonferroni that I illustrated earlier on. And the closed testing principle is an hierarchical procedure that allows you to reject an hypothesis only if the hypothesis that implies that hypothesis has been previously rejected. So in the case of this illustration here, let's say we have two null hypotheses, H1 and H2, okay? The intersection of those two hypotheses will imply H1 and H2. So the closed testing rule is reflected as we can only reject H1 at level alpha if the intersection hypothesis has been rejected at level alpha. That's what it translates to. So let's see some example here. Unfortunately, the Zoom, I don't know if I can move this, but I will move on in the interest of time. So this example, well, the bottom line, I think everyone can see, but let's say you have three hypotheses. You have three hypotheses. After conducting the analysis, the unadjusted p-values for the first hypothesis is 0.04. For the second hypothesis is 0.42. For the third, it's 0.01, right? If you are following a Bonferroni adjustment, what that means is that we're going to split the overall alpha equally across all three. That's what that translates to. And the graphical way to illustrate it is what I'm showing here. Now, if we apply this Bonferroni adjustment to the p-values that we have here, only the third hypothesis will be rejected. The others will not be, because the p-value is in excess of alpha divided by 3. Yeah, so the adjusted p-value in that case for the third hypothesis is simply 3 times the raw p-value, as I illustrated in the previous slide. And for the second, for the first hypothesis, you have 3 times 0.04, which is 0.12. And then for the second one, the minimum of 3 times this and 1. In that case, that's just 1. Okay. And if you compare all this adjusted p-value against 0.05, again, the only one that remains significant is the one for the third hypothesis. Now let's think about another procedure, the Holmes adjustment procedure. The Holmes adjustment procedure graphically can be illustrated this way. And this essentially let you go further from what you've seen with the Bonferroni, right? What this is saying is that after rejecting an hypothesis at alpha divided by 3, you are allowed to transfer the alpha on the rejected hypothesis to the other nodes, okay? So let's start from H1, okay? With you share a half with H2 and a half of that with H3 if they have not already been rejected. And the same goes for each of the node. That's essentially the Holmes procedure. And under this scenario, again, you will start with the node where you have a rejection first. And it works the same way like in the case of the Bonferroni. We are rejecting the H3 node first, the third hypothesis. And once that is rejected, this alpha divided by 3 can be floated into the remaining hypothesis, H1 and H2, which makes the available alpha there, alpha divided by 2 and alpha divided by 2, okay? Because of this, this allows you the floating of the alpha. You then calculate what the adjusted p-value is. In this case, since you only have 2 left, 2 hypotheses left, each being tested at alpha over 2, the reality is that none of these hypotheses can be rejected at alpha over 2. So you are not able to take out any of the nodes anymore, right? So even though this is a much more powerful procedure because of the ability to float alpha around, in this particular example, the result is not any different from the Bonferroni. The other approach is hierarchical testing. Hierarchical testing where in the design phase, we've already predetermined that we are going to have a primary hypothesis. And if that is successful, we go to a secondary one, the first secondary hypothesis. If that again is successful, we go to a third hypothesis. Preemptively, we are spending all the available alpha on the first hypothesis. If we fail to reject the first hypothesis, then none of the subsequent hypotheses can be rejected. This is a graphical illustration of this hierarchy. Using this example to walk through this again, in this case, because we are spending the whole alpha on H1, we are able to reject the first hypothesis. Then the alpha, the whole of the alpha, 0.05, is floated to the second hypothesis. And then we have to test this hypothesis at 0.05 level, which we cannot reject because the p-value is 0.042. So we can only reject the H1 hypothesis here, and we stop. So the adjusted p-value under this testing strategy for the first hypothesis is 0.04, and since it's less than 0.05, that's rejected. The remaining one are in excess of 0.05. The last strategy that we're going to consider here is what is called a gatekeeping strategy. Yeah, it's in some way it's employing flexibility. It's an adaptation of this Holmes type adjustment, where in fact you are again using element of hierarchical testing. You are spending all the alpha on the first hypothesis, and then when the first hypothesis is successful, you are moving alpha to the second family of hypotheses, which is comprised of two hypotheses, H2 and H3, at the same level. But at the same time, even though they are in the same family of hypotheses, the hierarchy is slightly different because we are floating 3/4 of alpha to H2, but only 1/4 to the third hypothesis, right? And once you are able to reject either of them, then you are able to float alpha in either direction. Now, in the case of this example, again, because you are using a gatekeeping strategy that allows you to spend all the available alpha on the first hypothesis, H1 is quickly taken out, and you are left with this simplified graphical illustration, H2 and H3, in which case what is available to H2 is 3/4 of the available alpha and 1/4 of the available alpha on H3. Okay. Now, when you float it as specified, again, for this particular example, you are able to take out, you are able to reject the third hypothesis. And then the remaining alpha, which is everything that you started with, is available for testing the second hypothesis. And in this case, you will fail to reject the second hypothesis because the p-value is in excess of 0.05. So you ended up, so the same data, imagine, but the multiplicity strategy determines what is successful and what is not. In the case of hierarchy, in the case of the first two methods, the Bonferroni and Holmes, the third hypothesis is rejected. In the case of hierarchical testing, the first hypothesis is rejected. In the case of the gatekeeping strategy, in fact, you ended up rejecting the first and the third. So the specification of your multiplicity strategy is very important. And that is why the context of the experiment, what you know about the setup, is what drives you to set up an appropriate testing strategy. And this is not what you come up with alone as a statistician. You have to collaborate with most often our clinical counterpart to decide on what is best for the situation at hand. So the example here that is being presented is in the disease called chronic obstructive pulmonary disease, COPD for short, where we have 4 hypotheses. And the reason for the 4 hypotheses is that there are 2 doses, so each dose is being compared to the placebo. And then you have 2 endpoints. The first endpoint is the annual rate of exacerbation. And the second endpoint is the forced expiratory volume in 1 second. The primary endpoint is designated as the exacerbation rate. So in this case, if you simply adopt a Bonferroni adjustment, a Bonferroni adjustment is not flexible enough to give you the context that one is primary and the other is secondary. A Bonferroni adjustment will simply divide the overall alpha by the number of hypotheses, in this case 4. We don't want to do that because it doesn't match the context. So the context, the relative importance of the hypothesis dictated by the setup is very important. So in this case, yeah, we have primary, but you are testing the primary on 2 doses, the low dose and the high dose. So these are the 4 hypotheses. One strategy is to split the alpha on the 2 doses, right? On testing the primary corresponding to the 2 doses, alpha over 2, alpha over 2. And then if you are successful on the primary, we'll float the whole of the alpha over 2 available for that dose to the secondary, yeah? And if you are successful on the secondary, you float the entire alpha to the other dose. So you can see how this can be more powerful than Bonferroni or even HORN because of this flexibility to float alpha around from one dose to the other. Once you are able to clear the 2 hypotheses on the dose. The alternative, there is also an alternative. Instead of floating the overall alpha 1 to the secondary hypothesis, you can say that I'm going to float half to the secondary and float the remaining half to the alternative dose. So what dictates this is the context of the setup. This is just an example. In summary, multiplicity is a very critical challenge in drug development and one that has to be dealt with in order to make good decisions. A decision that ensure that the risk of false positive is well controlled. There are different methods in the statistical literature on multiplicity control. Many research has been done in this area, and as statistician in drug development, one needs to be aware of all these strategies and their understanding in conjunction with the understanding of the context of the current problem would dictate which strategy one should go with. There are a number of aspects of multiplicity that we haven't touched on here, that we don't have the time to, but we haven't talked about the potential correlation between test statistics of different hypotheses. There are testing procedure that cater to this. The Donut test being an example of that. Dose response is another framework we'll be talking about, where you can factor in correlation across endpoints in dealing with the multiple testing problem. The other context is where you are in the group sequential testing framework. There's a segment of this workshop that is devoted to that later on. Many aspects where multiplicity can occur that we haven't dealt with here. So I will stop here and just some references on what has been presented, the graphical testing procedure, and also some other basic multiplicity testing approaches. So I will stop here. If we want to take maybe one or two questions before we move on, that's fine with me.
Let's appreciate Dr. Adewale Adeneye. We have retreated to another interesting topic, that is multiple hypothesis testing, statistical endpoints. We still have questions on sample size examination that was given to us by Dr. Smith-Brand, who is secret to such. And we have one or two questions here before we move to another topic. We can entertain that. I want to call on Professor Ugo to have his comment. Awesome.
Thank you very much, Dr. Adewale. I also enjoyed your presentation this time. It is very innovative. I think I'm just thinking about how to extend it to other areas of statistics, because we also encounter multiplicity of hypotheses in other areas of statistics. Please, I would like you to throw more light on the derivation of this new alpha when you have such multiplicity. You know, where all of them are being tested at the baseline of alpha equal to 5%. I want to see the standardization of the procedure for increasing the alpha value. Thank you so much.
Okay. Wonderful. Do we still have one or two comments? Online comments? Dr. Lee, can you take that? Can you react to that? Waiting for before we move to the next.
Okay. I think the question is about the derivation or the mathematical implementation of these different procedures. And depending on what it is that you're using, the derivation of, say, the adjusted p-value, as the route we often go through, will be different. Yeah. Since this is targeted to just being an introductory presentation of the topic, we didn't go into the derivation of a specific method, except the simplest case of the Conferoni adjustment, which is just scaling the p-value by the number of hypotheses that you have. And there are other cases where, in the case of the sequential testing as well, where it's very clear how it flows. But the mathematical derivation of the adjustment, they are well discussed in these references that I will encourage the audience to look at. Yeah. If someone is speaking in the room, I can't hear you.
I'm the one speaking.
Right.
Just for the benefit of our postgraduate students, and of course, a good number of our members that are running the postdoctoral researches in this area. So can you please identify recent research gap in this area of multiplicity testing?
Okay. Yeah. Thank you for that question. I would say that there are advances in the area. The graphical method that we talk about is one of the most recent approaches to multiplicity testing. In terms of research gap, I would say that the interaction of multiplicity when it comes to, say, multiple endpoints and group sequential method that we'll be talking about later on, when in the context of a clinical study, you have both of those dimensions going on. That's not within the scope of what I presented here. There's been some research in that domain as well, but it's still an evolving area where you have a group sequential design, and also you have multiplicity issues, maybe due to multiple treatments, or due to multiple endpoints based on which you want to evaluate the success or failure of the study. So those are areas that will benefit from further research. I haven't provided later on in what I will be speaking about in the group sequential testing framework. I will provide some references specifically for that. What I haven't provided in there is also the interaction of the multiplicity here and multiplicity as a result of group sequential testing. Those are additional references that I'll be happy to provide.
Thank you very much, Colin, for that insight. On this note, I think I'll move to the next. We have some online participants that want to ask one or two questions, but because of time, I want to take note of your questions, please. Just note down your questions while we have the next presentation. Then we can take the other questions alongside the other comments on the presentation. Dr. Rowan, who is taking us next?
Yes, that's mine as well. Okay. So interim analysis and group sequential design. I will want to start with a motivating example so that I can situate what the problem is. So this particular example is in the area of ophthalmology, where I work. Dry eye disease in particular. Dry eye disease is a multifactorial disease where the disease can come as a result of exposure to certain elements in the environment. It can be as a result of what the patient is predisposed to. Maybe they're just not producing the normal lubrication of the ocular surface enough. So there's deficiency in their aqueous layer. And this disease do manifest by ocular discomfort, general discomfort on the ocular surface. The patient can also be feeling some itchiness in their eyes. For this particular case, the focus is a clinical trial where a topical lubricant is being investigated against a placebo, yeah, so to say, and to mask the different harms so that patients do not know what they are receiving, or the investigators do not know what is being administered. There are a number of things that we followed using the previous principles we talked about. So the active drug is being dosed 4 times a day. Placebo is also being dosed 4 times a day. So you don't want to create a difference that will make it obvious which group is taking what. And the efficacy endpoint is based on assessment of what is called tear film breakup time. In this case, I will say that if the ocular surface is healthy, if a patient doesn't have dry eye, then when you lubricate the ocular surface, it should take a long time before the tear film that is covering the ocular surface to break up into chunks. Yeah, so this type of testing is used to assess the health of the ocular surface in this particular case, tear film breakup time. Okay, and in this case, it was assessed at baseline, and at the end of 35 days of continuous treatment, we assess tear film breakup time again, and the endpoint is changed from baseline in tear film breakup time. In this case, in planning the study, we have to think about what we know going into the study, because there are certain assumptions that we have to make. In particular, the magnitude of the treatment effect. We don't know. There's uncertainty because there were early studies that indicate that it could be small, it could be large. There's one study that says it could be about 1 second for the breakup, for tear film breakup time to happen. There's another study saying that it could be twice as big, 2 seconds. Yeah. So how do we handle this situation? How should we design a study to establish the superiority of the topical lubricant versus placebo? Yeah. In this particular case, we followed a group sequential design where we powered the study for a small treatment effect, and we incorporate an interim analysis when 35% of the total planned sample size have attained the primary endpoint. And the hope is that if in fact the treatment effect ended up being larger than what we planned for, then you have sufficient power to stop the study at the interim analysis. Okay? And in this particular case, it happened that at the interim analysis, we have generated sufficient substantial evidence that the treatment is efficacious. And so the trial was terminated based on the interim analysis. So how do we set this type of analysis up? And what is an interim analysis to start with? An interim analysis is an analysis that allows you to inspect accruing evidence. You are not at the end of the trial yet. The trial is still ongoing, but based on intermediate evidence, you want to evaluate the evidence of efficacy. You want to test the primary hypothesis for the study based on accruing evidence. And so we will go ahead in that case at the interim analysis to break the blind. In ophthalmology, we also call that unmasking. That means you are revealing the treatment code so that you can facilitate the necessary analysis. Why would we want to do this? Interim analysis are relevant to stop a trial early, either because there's a safety concern, right? Even though you've done all the earlier phases of study, or you've done studies that shows that there's no safety concern. Remember, safety, you start from, even from preclinical and continue to evaluate. Safety concern can emerge at any point during development. So a trial can be stopped at interim due to safety concern. It could be stopped due to overwhelming efficacy. In the case of the example that I used to motivate this is the scenario. We've generated substantial evidence of efficacy. The third possibility is when we are evaluating lack of efficacy. Drug development, it costs money. It costs millions of dollars to conduct these studies. Sometimes we want to protect our investment. We don't want to wait all to the end to find out that something is not working. So we want to build in interim analysis to assess whether it's meeting the basic criteria that one would expect if it's doing what it's supposed to do. Also, we can conduct interim analysis to optimize the design, to modify certain aspects of an ongoing study. We can also conduct interim analysis sometimes as a means to use to plan other studies. So this is often called an administrative look. It has nothing to do with this ongoing study itself, but you want to use the information somewhat to inform other studies in the development program. So type 1 error rate is very critical that we control. Just like we illustrated the other time, that when you have multiple hypotheses, each of them being tested at level alpha, in this case 0.05, if you do as many of those, it's almost a certainty that you will have a false positive. So the same applies to interim analysis, that you have an inflated type 1 error due to multiple testing. In fact, a simulation was done by Tiatis and co in the Start Medicine population in 2006, where the same phenomenon was illustrated. Yeah, this is a setup where each sequential test is being tested at level alpha, say 0.05. If you just do one test, you control as expected at 0.05. But if you do two, you see how quickly, with increasing number of repeated look at the data, how quickly the false positive rate can increase. So to set this up, let's, even though this can be extended to other setup, but just for ease of discussion here, we're going to focus on a superiority testing situation where we have an hypothesis where we are testing the null hypothesis delta equals 0 versus the alternative that delta is greater than 0. And we're using a one-sided level test at the level alpha. The power is desired to be at 1 minus beta, where beta is your type 2 error, as illustrated earlier on. And we want to be powered at 1 minus beta, while testing at level alpha, assuming that the expected treatment effect is delta 1. This is the information that you need with the assumption of if this is a continuous endpoint, for example, with the assumption on the standard deviation as illustrated in Brian's presentation earlier on, determine the sample size. And now what you are introducing is that instead of just having one hypothesis, you want to do K-1 interim analysis, which correspond to K-1 different hypotheses, including the final analysis, which translate to a total of capital K hypothesis. Yeah. Now, in this framework, depending on the nature of the endpoint, how you monitor information could be different. For example, for normal and binomial endpoint, with the sample size is just the measure of the information. So the maximum information possible is the required maximum sample size. It's straightforward in that case. For time to event endpoint, it's not so much the sample size, it's the number of events that determines the amount of information. So the required maximum number of events is what drives the power for the study. In this case, we're denoting it by D max. In general, the Fisher information is used to quantify available information in a group sequential framework, which is often estimated by the square inverse of your standard error. Yeah, and the other notion I will introduce is the notion of information fraction, which is essentially the ratio of the information available at any point in the analysis to the targeted maximum information planned for at the beginning of the study. Here, I'm introducing the notion of the test statistics that is used for testing. So at each interim analysis, let's say that you're testing at the point where you have information fraction Tj. The Z statistics at that point is just the estimate of the treatment effects divided by the corresponding standard error. And that's the typical word statistics. It's been established to be an efficient estimator. And it can also, if I write, you remember on the previous slide, we estimate the maximum information by measure or by the square inverse of the standard error, which can be calculated at each interim analysis. And so if you define the information available at any particular analysis by this, we can rewrite the word statistics as a function of the information fraction. And then to go further into the setup of this, the distributions theory is set up such that we work with a variable defined based on the word statistics. So the square root of the information fraction multiplied by the word statistics. And these W's, they have asymptotic normal distribution with mean defined by eta ti and the measure of the standard variation based on the notation that we are adopting is simply the information time. And if you're looking at 2 time points where TJ1 is the earlier information time, because you are using the same patient population, there's correlation between the 2 test statistics corresponding to the 2 information time. And because of the setup, that covariance is simply the earlier information time, TJ1. And also the 4th assumption here is the notion of independence, where you have the information, I think it's easier to illustrate as the information gathered between the successive intervals. So this is the first analysis, TJ1. Then if you're looking at the W statistics computed at TJ1, and the difference between the test statistics at the 2nd time point and the earlier time point, those 2 parameters are independent. And so with these 4 conditions, it has been established that with all this, you can compute your group sequential stopping boundaries and essentially determine the crossing probabilities at each analysis time point. Again, how do you compute the efficacy stopping boundary under this setup? So we're assuming that we have k time points corresponding to this T1, T2, Tk information fraction or time point. The one-sided stopping boundary are constant, C1, C2 to Ck, such that the trial stop whenever the test statistics cross the boundary, the corresponding boundary at any of the interim analysis. So in order to control overall type 1 error, what we are doing is that under the null hypothesis, the probability of crossing the boundary at any time point, we want it to be equal to alpha. So the problem just becomes choosing specific constants such that this probability is not more than alpha, okay? And it doesn't have a unique solution. There's no unique solution to this. There are many CKs that can potentially satisfy this condition. And so the question is, how do we choose this boundary constant? The earliest proposal in group sequential design are the Pocock boundaries as well as O'Brien-Fleming boundary. Pocock proposed to use a constant boundary regardless of the time points, regardless of the number of analysis. We want it to be constant. O'Brien-Fleming recommended a variation of that which increases, actually in this case, we will say that it starts by actually reducing the likelihood of crossing the boundary at the earlier phase of the analysis, where you do not have as much information accrued yet. So the boundary will be high in that case. And as you accumulate information, the boundary statistics gradually decrease. Wang and Stiestich proposed a family of boundary which has Pocock and O'Brien-Fleming boundary as special cases. So for example, if you look at this boundary and say delta equals half, essentially that takes you to the Pocock. Yeah, if you're using delta equals 0, that gives you the O'Brien-Fleming boundary. Again, it's the same thing. You have to compute the C here, the constant C, such that this condition is satisfied. And this is done through recursive integration. And the other point to be made here is that this earlier approach to interim analysis using these boundaries, they require that you have equally spaced analysis time point. So if you're going to have two analyses, one interim analysis and a final analysis, they require that you have, say, at half in the middle of the trial, where you have half of the information, and then the final when you have all the information available. So it requires evenly spaced analysis for their validity. Land and Demet introduce what is known as the flexible error spending approach that allows flexibility with respect to the timing of the analysis. With the contribution of Land and Demet, we can conduct analysis at non-evenly spaced time points. We can also adapt the number of analyses that has to be done. If we do that with any of the earlier boundaries, we will violate the condition for type 1 error control. But the approach of Land and Demet provided additional flexibility. So the notion that was introduced by Land and Demet is that you define a spending function, say alpha of t, a function alpha of t, and it's a spending function because at time 0, at the beginning of the trial, you haven't spent anything. Your alpha is 0. But at the end, when you have moved through the information scale, you are at the end of the information scale, you've exactly expended the allotted alpha. So any function that satisfies this condition is a spending function. It's a monotonic increasing function, alpha t. And the next step is that in determining the corresponding boundaries, the C1, C2 to Ck constant, you have to solve this overall equation, which is solved in a sequential fashion. You start with the force analysis, yeah, which again, you have to look at that in force interim analysis. What is the information fraction at T1 at that analysis? And when you put T1 in your spending function, it gives you the targeted alpha to be spent at that force analysis. And to control type 1 error at this level, at that force analysis, essentially is to ensure that under the null, the probability of crossing that constant C1 is exactly alpha of T1, okay? And if you fail to reject the null hypothesis at the first analysis, then the next step is that in the second step, the alpha spending again correspond to alpha T2, and that has to control the overall probability. So this is the sum of the probability of crossing the boundary at the first analysis plus under the null, the probability that you did not cross the boundary at the first analysis, but you are crossing at the second analysis. Okay, so this is this equation is solved in a sequential version, depending on the number of analyses that you have in total, to ensure that you have overall control at the desired level alpha. So that's the whole approach for flexible spending approach. And in what Lan and Demet propose, they provided special function that corresponds to an approximation of the traditional Pocock and O'Brien-Fleming boundaries. Here is a case where you are more aggressive, the Pocock boundary is more aggressive, it spends the available alpha very quickly. The O'Brien-Fleming type boundary, on the other hand, based on what we discussed earlier on, the chance of rejecting the null hypothesis is small at the beginning, and as you accumulate evidence, you increase the likelihood over time. So it's known to be conservative. In this, it conservatively spend the allotted alpha. So if one is looking for a spending function that is close to a fixed design type approach, the O'Brien-Fleming type of flexible function is encouraged in that case. So let's go back to our earlier example in this case. So it's a superiority testing. We want to establish that the topical lubricant is efficacious in treating dry eye, and that we want to establish by looking at change from baseline in tear film breakup time. In that study, the design, the randomization ratio for the design is 1:1, and we are targeting a one-sided alpha. We want to control type 1 error at 2.5% one-sided. The targeted power is 90%, okay? The assumption here to start with is that the expected treatment difference is just slightly under 1 second. And the standard deviation for our change from baseline in tear film breakup time is 2.5 seconds. Now, what we're looking at is different design options. The first one is the fixed design, where we are only doing one look at the end of the study. We don't want interim analysis in this case. Okay, for that one look design, there's no need to stipulate a boundary. The only boundary that is relevant is the alpha that is available at the end. Yeah. So information fraction at that end is to have the full unit of information. That translates to a maximum sample size of 295 subjects. The second alternative here is a design where we are using the Lan and Demet approximation to the Pocock boundary. And here we are actually conducting the analysis when let's say 40% of the patient have had the primary endpoint of interest. And the final analysis, if we proceed forward, will be at the end. Okay, in that case, because you have two opportunities to reject the primary null hypothesis, because you have two opportunities, the Pocock approximation of the length and demand using the length and demand spending function translate to a maximum sample size of 324. That means in order to maintain your power, given the multiplicity adjustment that is necessary to maintain alpha at the desired 2.5%, the penalty for that is an increase in sample size. Okay. On the other hand, if we want to use the O'Brien-Fleming type spending function, which is known to be conservative, meaning that it doesn't spend much alpha at the earlier phases of the analysis. In that case, in fact, you don't see an impact in this particular example on sample size. It stays the same. That's not always the case, but the point to be made here is that the O'Brien-Fleming type spending function tend to closely, it's usually close to what you expect under fixed design because the conservative nature of spending your alpha in that case. So again, for this particular example, the C1 for the Pocock type boundary is 2.224. So remember, I'm using 2.5% significance level. So the corresponding test statistics for the critical statistics for that is usually 1.96. So in this case, because of the introduction of interim analysis, instead of 1.96, that has increased to compensate for the fact that we are doing interim analysis, and that needs to be accounted for in order to control overall type 1 error rate at the desired level. So instead of 1.96, you see it's inflated. The same thing at the final analysis. Remember, this is just the land and demand approximation of the Pocock boundary. If we were to be interested in the original Pocock boundary, then in that case, the analysis must be happening at exactly 0.5, right? And in that case, also the critical cost, the constant will be the same level. Here it's about the same, not exactly the same, okay? On the other hand, the corresponding boundary for the O'Brien-Fleming type spending function, you see it's very high here. You will have to have overwhelming evidence of efficacy in order to reject the null hypothesis at this first interim analysis. If you do not reject, you see that you almost have all the alpha available translated into about the same critical constant as you will be using for the ordinary analysis that doesn't have interim analysis. So in this particular case, some of the things to look at are the exit probabilities under different conditions. So under the null hypothesis where delta is equal to 0, what we want to see is that the cumulative probability of exiting and rejecting the null hypothesis at any analysis sum up to the desired alpha, which is the case here. Yeah. Under the assumed or expected effect, we expect the sum of the exit probability to equal the power. Okay. Remember that in the setup of this particular example, we said there's an earlier study that suggests that that magnitude of effect can be as high as 2 seconds. If that is the case, then if we conduct an interim analysis at 40% information fraction, we have about 84% power to stop the analysis at the interim analysis. And that's why this approach is attractive under this scenario. So in the case of the particular example that I've been talking about, the dry eye study, we use the land and demand approximation of the O'Brien-Fleming boundary, where the boundary p-value is almost close to using nothing at the beginning, right? And then you have almost the entire significance level for your final testing. That is the plan. In actual fact, before we could conduct the analysis, even though we planned for 40% information, the information available, the sample size that has been accrued as at the time of the analysis, translate to 47%, right? And because we're using a flexible alpha spending approach, we were able to adjust appropriately and compute the appropriate boundary that corresponds to the available information fraction at that time point. Yeah, and we ended up observing a difference of mean of 1.3 seconds, and the corresponding p-value is 0.006. And when you compare this to the boundary that correspond to the available information fraction at that analysis, we are clearly rejecting the null hypothesis in favor of the superiority hypothesis. So the trial stop for efficacy at interim analysis in this particular example. In summary, interim analysis should be carefully planned and it should be carefully documented to ensure that there's no unauthorized access to data. We want to ensure that the trial, there's integrity to it. So when we have interim analysis, we don't leave the treatment code accessible. In fact, it's not accessible to the treatment, to the study team. And that's true for all our clinical studies, regardless of whether it has interim analysis or not. But when it has interim analysis incorporated, there's a process whereby the treatment code is released to the team that is charged to conduct that interim analysis according to the stipulation of the protocol. So you conduct interim analysis like we talk about in the group sequential framework. There are other approaches known as adaptive design framework in general, where you are not just about stopping the trial for one reason or the other, but you want to modify the characteristics of the trial going forward after that interim analysis. Group sequential design in particular allows you to do early stopping, and early stopping can be for multiple reasons, as we already discussed. And if appropriately done using the available methodology, it ensures that type 1 error is appropriately controlled. In practice, as I said, we don't use the traditional O'Brien or Pocock boundary. We want flexibility and to ensure that the analysis is valid. We typically use the flexible alpha spending approaches. So I will conclude by saying that the planned information fraction, as we just saw in the example that we illustrated here, you don't always achieve the exact thing, but it's very important that whatever information fraction you have available, there's a way to translate that in a way that ensures overall control of type 1 error. Thank you. Some references here on this topic is a well-researched area in pharmaceutical drug development. I'm happy to take a few questions.
Thank you very much, Dr. Adeni, for this beautiful, another beautiful presentation. And we appreciate Dr. Adeni. We please appreciate this scholar from that hand. Thank you very much for this. This is an opportunity for us to ask questions and interact meaningfully with the presenter. We are treated to a really nice topic. Actually, it's supposed to spawn a lot of questions in terms of the direction. And of course, there could be people among our participants that are actually researching this in this area of the clinical trials and other associated issues. So this is an opportunity for us to raise one or two issues. And on the earlier presentation too, there are some people that have indicated interest to ask questions that we couldn't take. So this is an opportunity again to visit that before we move to the last presentation for today. So any query? Maybe I start from my head here. Dr. Adewale, thank you very much for this presentation. Just going forward, I look at for example, the interim analysis and the head of the interim analysis. One of the objectives of engaging in interim analysis is to ensure that if the treatment effect is futile, there's no point going further, wasting resources when actually what we wanted to determine is not really the case. So, given that, is there any, will you suspect any overlap between this interim analysis and the phase 2 clinical trial, for which at that level 2, you just look at 2 cases and to determine the possible effects of the treatment before you move further? But certainly whether the intervention, whether the drug or the treatment, is actually what is expected of it to do before you move to the next phase. And this is the actually one of the areas to achieve similar objectives. Is there anything you are suspecting in terms of overlap?
Yeah, so Prof, I didn't hear all the comments or questions very well. It's hard to hear.
Well, you presented a nice topic on the interim analysis. And one of the objectives of carrying out interim analysis is to ensure that when you finish the study, you determine whether the impact of the treatment is actually giving you the result that is expected, or is going to give a futile result. If you're going to give a futile result, you stop at the early stage instead of wasting resources on the. So, and we look at the phase 2 clinical trial too, for which at that phase 2, you recruit few subjects into the study to determine the possible effects of the drug or the treatment intervention. Okay, I'm not asking, do you suspect any overlap between these 2 concepts, the phase 2 clinical trial and interim analysis?
Oh, okay. Thank you. Yeah, so Actually, I don't want to mislead anyone that what I presented is exclusive to phase 3. It's not. It could be used at any phase of clinical development, especially maybe I should say phase 2 and 3 for the most part, but also in phase 4 as well. I'm excluding phase 1 because very often the objective there is assessing safety, not efficacy. So with that, the methodology can be applied even in phase 2 development as well, where you want to for the same reasons that I talk about here. But something else that you alluded to is the angle of futility, right? I only briefly alluded to that in my presentation. I didn't go through the methodology around that necessarily. But that's also part of what can be incorporated into a group sequential design framework. In fact, you can have two type of boundary concurrently in the same study. You can have an efficacy boundary, which you are using to determine success, but also you can have a futility boundary that if you cross that as well, you are declaring futility. Yeah. So in the case of futility, instead of defining an error spending, an alpha spending function, like I described in the presentation, what you are describing is a beta spending function because what you want to control there is your type 2 error. The risk of a false negative is what you are controlling with futility. And it's well established in terms of how you do the computation between those two boundaries to ensure that overall you meet your type 1 error control requirement. But in phase 2, again, we have more flexibility in terms of how we approach this. Yes, at any stage, we want to ensure that we are making the right decision. But in terms of the methodology or the statistical tool available to us, there is more that can be done in phase 2 than phase 3. Even though the statistics apply equally to both, but when it comes to phase 3, the regulators have certain preferences to ensure that they can admit the evidence that is being generated as a basis for registration. I hope that addresses your question. If anyone is speaking in the room, we can't hear online. Any other question? We still can't hear you.
Oh, okay. Thank you, Doc. This is, I want to commend your presentation and I enjoyed every bit of it, especially the mention of the alpha spending function. It's something that is very, that I need to read about. But as you describe it, I'd like to know, do we have any relationship around this function that suggests that it operates like what we know from the crossover designs? Okay, from the crossover design, do they have any relationship? because the way you describe it, I could link it to some of the things I know in crossover design. Then two, when you were talking about the CJ equals to C, I'm talking about the monotone increasing function. Is it almost surely, because you said any function that exhibits T belonging to a closed band of 0 and 1 is a monotone increasing function. Yeah. I seek clarification on that because I believe that it can be opened. It's open to these closed bounds when we talk about monotone increasing function, because it cuts across disciplines. So I want to ask whether it is in all cases that you are referring to, or this specific, or you are talking about this specific situation. Then in the expression you gave for CJ, where you say CJ equals to C divided by the square root of TJ. If the square root of TJ goes to 1, that is when CJ equals to capital C. Do we have a practical situation where you likely suspect that the square root of TJ will approach 1? So that is the question I have. Thank you.
Okay, maybe I can start from the last one. So the square root of TJ, you remember T is the information fraction. So at the end of the study, for the final analysis, that's always 1, right? You have the full information that is expected from that study at that time point. Yeah. And then if I go to your second question about monotone increasing function, yes, any monotone increasing function that satisfies those boundary requirements, meaning alpha of 0 equals 0 and alpha of 1 equals alpha. If it satisfies those boundary requirements, is an admissible spending function. Now, whether or not it could be used depends on the behavior, right? So people have defined different spending functions in the literature. I only touch on the Land and Demet approximation to the Pocock and to the O'Brien-Fleming boundary. There are other spending functions that has been described in the literature. And what is common with all of them, there are different mathematical forms of those spending function. What is common to them is that they all meet that definition, right? But their behavior will be different, and whether or not they can be used under any particular application depend on the context. What do we want to achieve with the problem at hand? Then your first question about the relationship of group sequential design to crossover design. So when you talk about crossover design, you can contrast that with a parallel design. But the hypothesis testing, the hypothesis to be tested can be the same, right? In a crossover design setup, you can be testing for superiority, the same as in parallel design, right? So the mathematical setup is the same as long as for that crossover design, you are also introducing the notion of interim testing or group sequential framework, whereby you are doing interim analysis and you want to understand how to accommodate for the multiplicity issue that arises as a result of the multiple time assessment of the data. So the problem is the same. The example that we've used here just happened to be in the context of a parallel design, but the same translate to crossover design or any other type of design for that matter. The important thing is the definition of your hypothesis. And this framework is just helping us to determine how we want to sequentially test that hypothesis. The design can be anything that suits the situation.
Yeah, thank you very much, Doctor, for this beautiful insight. There is a general request by our participants, both online and people that are in the institutional room here. And the request is directed at you and other presenters that for you to avail us the opportunity of having the materials presented, shared to us so that we can leverage on that for better understanding. I wouldn't know whether what your response will be to this. Thank you.
Yes, I think that's fine. It's fine with all the materials, and I think Oliver and Brian will agree to that.
A round of applause to our facilitators, please. We will be sharing the material with us by the time we close the session. So in a quick succession, shall we move to the next presentation? Dr. Oliver Sander is waiting for us. And I want to put our spot.
So Oliver, go ahead and share it. And we'll be handing back and forth until the end.
Okay, so let me just share my screen.
Share screen, please. Thank you. Go ahead, please. Thank you.
So can you see the presentation?
Yes, we can. Please go ahead.
Okay, great, thanks. So in this session, Brian and myself will cover clinical pharmacology, pharmacokinetics, pharmacodynamics modeling, and dose-finding studies. And so in particular, we are looking at questions that are addressed and approaches to address these questions in the somewhat earlier and like middle phases of drug development. And so we're looking at clinical pharmacology, which you could kind of define as studying the proper use of medicines in humans. So it's essentially trying to find out how to use the medicines in practice, in clinical practice that we're developing. We're looking into dose finding, which is trying to figure out at which doses we should use the drugs. And we're looking at pharmacokinetics, pharmacodynamics, and quantitative models to describe pharmacokinetics and pharmacodynamics. Kind of taking a step back, and Nia had already at the beginning, like briefly kind of hinted at this, but kind of again taking a step back at like what we're really trying to do. You can really think of drug development as going from here left-hand side from a chemical substance. So you have some substance where you have some hint or some hope that this might be useful to treat a certain disease, and this is the starting point, and you want to go to the right-hand side, which is a drug label and a package insert for a drug, which essentially describes in detail how you use a medicine in patients, and then even how to use it in different patients. And so the question is, how can you move from left-hand side, so like really just a chemical substance or a molecule, to really a medicine that you know exactly how to use? Looking in some more detail at this label and package insert, and then I'm sure like many of you are kind of aware of this and then have seen this as it's like most drugs that you use, they have these package inserts bundled with them. And so this here is for Coartem, which is the antimalarial drug that Nia had already mentioned at the beginning. And if you look into this package insert and the label, what you see there is quite detailed description of actually how to use the drug in patients. So for example, here on the left-hand side, there's a whole section about dosage and administration. So this tells you in detail how to take the drug, at which dose, so how much of the drug you take, at which regimen, so for how long and how often you take the drug, and in this case here for Coartem, what the dose is that you should take based on body weight. So as you see here on the left-hand side in this table, for Coartem, it depends on the body weight of the infant, the children, or the adult, how many tablets per administration the patient needs to take. And on the right-hand side, you see, sorry, let me just, so the zoom window is on top. Okay, on the right-hand side, you can see like some further information, like information on drug interactions. So for example, can I take this drug together with other drugs? And is there anything I need to take care of that? For example, if I'm taking, so as you look here, like a certain class of drugs called like CYP3A4 inducers. If I take antiviral drugs, if I take contraceptives, for example, what do I need to take care of? Or what do you need to be cautious about when I take other drugs in combination with this drug here? And if you look further at the bottom in this kind of table of contents of the drug label, there are a lot further points listed. So for example, how to use the drug in specific populations. So, and then this is here the point 8 at the bottom. So he has listed pregnancy, lactation, females and males of reproductive potential, pediatric use, so what do you need to take care of if you give this to children, geriatric use, what happens or what do you need to be aware of if you give it to elderly patients, what do you need to take care of if you give it to patients with either liver or a kidney impairment or disease. So like all of these information, this is something that you need to be able to put into this package insert and drug labeling. And so therefore, at some point during your drug development, these are all questions that you need to somehow figure out. So these are all questions that you need to explore as part of your drug development. And so again, coming back to these different phases of drug development, and again, Nii had before shown like a similar chart with different development phases. There are different questions throughout these phases that you try to address. And so you're kind of starting by, in the clinical phases, like what doses are safe. And so Nii has talked about the MTD, the maximum tolerated dose or tolerable dose. So like this is one of the first questions you're trying to figure out what doses are actually safe. Then in the next phase, you try to figure out, well, is there actually a meaningful effect at a reasonably high safe dose? And then this is typically done what is like called the phase 2B studies, then trying to really understand how is dose and response related, and therefore then being able to decide which doses and which dosing regimens do we want to take forward into the large phase 3 confirmatory studies, because these are the doses that we think are the right ones for patients, for which you want to prove that they're efficacious and safe. And throughout this whole process, there are these other questions that need to be taken care of, like what about special populations? What about other circumstances like taking other drugs, elderly patients, patients with impaired kidney or liver function, or for example, what about food effect? Is there anything you need to take care of if patients are taking food before or after taking the drug? And so how do you address these things? And this is what Brian and I will cover a bit in this section here. And with that, I'm going to hand over to Brian.
All right, just go over to the next slide, Oliver. Okay, so just before we get into this, just a reminder of the harsh realities in drug development by the numbers. 10% of our molecules tested in humans become useful medicines. It costs $2.6 billion is the amount of the average R&D spending for a useful medicine. Our patent length is 20 years for a new drug. And it takes approximately 10 years of that 20 years on average to develop the useful medicine. All right, next slide. And just seeing how hard this is, this kind of gives you a feeling, you've seen this before, but just how there's attrition of our drugs as we move down the drug development pipeline. Next slide. And the thing is, it's not getting easier. It's actually getting more expensive to do drug development in general. And you can see this in time. This is probably due because some of the easiest targets have already been figured out, and we're getting into harder and harder things. Next slide. One of the things is, and I mean, this Paracelsus goes back to 1493, right? And he made the statement, the dose makes the poison, right? And basically what he's saying is everything is dangerous if you take enough of it. The higher the dose, the more effect, both positive and negative, and understanding how to dose increases the value of a useful medicine. So one of the key components is that it's good that we know what medicine to use, but we actually have to know how to dose it to the individual subject. Next one. So if you think about all the ways we could try to increase the values of our molecules or increase our value, I said that 10% of the molecules are tested in humans becomes useful for medications. We could try to increase that chance of success, but I've been doing this 25 years or 30 years, and that has not budged. We could cut cost, but we try. But both ethically and regulatorily, there are things that have to be done. Last thing is we could get faster. We try that too, but some things take time. But one of the key ways that we can increase the value of a medicine is really to understand how to dose it in the patient. Next. So just to kind of give you a feeling for how a drug works, and this is going to be important later on as we think about this. So you can sort of think about this, and this is called a lock and key approach, but basically there's a receptor of some sort, and there's a drug of some sort. And the drug is floating around and it's locking to the receptor, which is causing some sort of an effect, right? That's what we're doing with drugs. The more of it, the more the drug you get, the more receptor is bound up, the more effect that you have, right? Next slide, please. And so a simplistic way of thinking about this, there's a hypothesis, or it's a known, that by attaching to a particular target, good things happen. There are a lot of targets in the diseased person. The more circulating drug, the more targets are engaged, more good things happen. Drugs, although designed to avoid, can also attach to other targets where bad things happen. You may conclude, if I know the amount of drug to a given person, I can optimize the benefit while minimizing the rest. Next slide. Yeah and no. The thing is that people are different. It's obvious, I'm sure it's obvious to any of you, a 135 milligram man might need more drug than a 45 kilogram woman, right? There's just more of them, so they need more amount. What they really need is that their concentrations be equivalent to each other, right? So the conclusion, since drug travel basically through the body, are interested in the drug concentration. It's not just weight that affects somebody's concentration, though. There's all kinds of things, right? The gender can, the age can, ethnicity can, genetics can. What other drugs one takes can. What and when you eat can affect it, and the nature of either that disease or other diseases can impact what the concentration is. And so we need to understand all those things. Next. So pharmacokinetics is what the body does with a drug. So if we're going to understand concentration, we need to understand what the body does when we take a drug. And there's actually 4 components. Sometimes we say ADME, but it's basically we take a drug, it's absorbed into the system, it's distributed throughout the body, it's metabolized. Metabolized is mostly, but not completely, done by your liver. Your liver's there mostly for poisons, but basically metabolism is sort of like your body's way of turning that compound into something else so that the drug can be eliminated from the system. That's sort of the idea of what happens to a drug when you take it. Next slide. So we measure the process with concentration, And here you see, although these are mean plots of concentration over time, any individual would have some sort of thing that looked like this. If we gave them drug, we could measure their concentration over time, and what you would see is that their concentration would go up until it peaked and it would start to eliminate, right? Two kind of statistics that come out of this for an individual is the area under the curve and the maximum concentration. And there are other things that can be studied, but I'm just going to concentrate on those right now. Next slide. So just keep in mind that getting the right dose is a long and iterative process. We actually have chosen to start at the end. We've sort of got the dose, right? We've got the dose that we're going to use in phase 3, which our goal is to get to a one dose. We don't always do this, that by the time we get to phase 3, we can give one dose. But even if we have that dose, we still have to understand whether or not I can give that dose to everyone or under all circumstances, or whether I can fill in that information in the label that we were talking about. So that's what I'm going to talk about right now, and then we're going to backtrack into getting the dose right itself. Next slide, please. So there are two ways for us to study trying to get the, trying to figure out what sort of things impact the dose. And this all has to do with concentration. There's experimental ways, that is, we can do studies, or observational ways, right? The experimental ways, the characteristics of these studies are usually their subjects are often healthy, or not a class of subjects that are in phase 3. And also the trials often can be crossover studies, and usually the primary endpoint has to do with pharmacokinetics, the AUC and the Cmax, for instance. Typical types of studies are food effects studies, drug interaction studies, renal impairment studies. So if you don't have a kidney, for instance, you're going to get rid of the drug differently than someone that does have a functioning kidney. Observational, these are characterized by, these are subjects that come from phase 3 studies themselves. So we can typically examine, though, differences in body weight in that, or effects on gender, or effects of age, if that makes any sense. It's observational, it's there. If we get the pharmacokinetic information, the concentration information, we can determine these things. Next slide. So in both situations, some basic statistical considerations. It is accepted, and if you've seen concentrations themselves, they tend to be log normally distributed. They're very skewed to the right, and so we accept that throughout with dealing with this sort of data. Now, also often the scientific interest is not in if two treatments are different from each other, but whether they are clinically similar. That is the examination of equivalence. And one of the ways of doing this is you set up an equivalence range, x to 1 divided by x. A conservative range is 0.8 to 1.25. And now you're interested in seeing if the ratio of the concentrations is between 0.8 and 1.25. That's typically done with a confidence interval, typically a 90% confidence interval, and you're seeing if it's contained in that range or not. And if they are, then you consider the two treatments, the two things that you're testing, equivalent to each other. Next. All right. I think I remember someone saying they worked with crossover studies. There's an amazing precision gain from using subjects as their own control. We don't do it very often in the pharmaceutical business because of the length of the trial for a subject. So many of the studies that Nee was talking about, they're just too long to consider crossover as a component to them, right? But you're also, the other component is that you always worry about a carryover effect, right? That in essence, will you have long enough washout period? Now, I don't have these concerns in pharmacokinetic studies. And the reason is the pharmacokinetic properties can often be measured from a single dose. So the studies are can be very short in nature. And as long as the drug is washed out, there's no carryover effect. And in fact, an easy way to check is to measure the pre-dose concentration right before the second dose is administered. If there's no drug available, if there's no drug there, if there's no concentration there, there's not going to be an impact on concentration in the second period. Next slide. Okay, so I talked about renal impairment, renal impairment. That's a study we do where we bring in a bunch of individuals that have renal disease and figure out what their pharmacokinetics are. We might compare them to healthy subjects. Obviously, there's no randomization there. It's almost observational. It's a study. We give a treatment, but basically, we're not doing any randomization. We're just comparing the results. You may want to control individuals in this way through matching them. So, for instance, when you bring in a renal subject, you might want to find a healthy subject that has a similar age, gender, body weight, sort of thing in order to match so that you have a comparison that makes sense. Next slide. Now I'm going to switch though. Those are kind of the experiments that we can do. Mostly crossover, right? Relatively small studies. These studies usually we can do with 25, 30 subjects in general, sometimes less. Population pharmacokinetics, I'm going to give you a little bit of a preamble to it. So far, I've talked about AUC and Cmax, and they're non-compartmental, and don't worry about that term right now, ways of, or model-free ways I can determine those. AUC can be found with something called a trapezoidal rule. I connect all the points, I find what the area is for that person, right? The Cmax would be the highest observed concentration for that subject. Now, it turns out though, basically, that you can describe that curve that I just showed you, where it goes up and it comes back down very well with something kind of compartmental model. And what I'm showing you is the equation for a one compartmental model with oral absorption. The concentration at time t is equal to the fraction of the dose absorbed times the dose times the absorption rate divided by the volume of distribution. The volume of distribution is basically how much space the drug has to go, minus the difference between the absorption rate and elimination rate, times the exponential of the elimination rate over time minus the exponential of the absorption rate over time. If you have dense sampling from an individual, there's no reason to risk improper model. Non-compartmental modeling is done. But if I don't have really dense information, a lot of concentrations from an individual, I can use this model. Next slide, please. So I want to do some math really quickly, though, just to show you that I can figure out what the AUC is of this model, right, which I said I was interested in. It just turns out I can integrate this over time from infinity to 0 and find out that the AUC is equal to the fraction of absorbed times the dose divided by the volume times the elimination rate. Okay, and I'm going to define a concept called clearance, which is just the fraction of absorbed times the dose divided by the AUC. The reason I've done that, well, the reason that I like this as a statistician, it gives me a metric to a certain extent that's very much like the AUC that I can use in the compartmental setting. But clearance has a mechanistic meaning among pharmacokineticists, but we can all think of it as kind of a reciprocal of the AUC if you want to think about it in that way. It's an AUC metric. So more math, if I set clearance equal to F times dose divided by AUC, we find that's equal to the volume times KE. And so if we let KE equal to clearance divided by volume, we can rewrite our equation that we just had. Instead of in terms of Ka, Ke, and V, we can write it in terms of Ka, clearance, and volume of distribution. And I'm going to do that because in general, pharmacokineticists feel there's mechanistic meaning in clearance. I kind of like it because clearance then becomes kind of a dose normalized version of AUC, and that's kind of helpful. Next Next slide. All right. So I've told you that we're going to get to a situation where we're going to use individuals from phase 3. We don't have dense sampling from them. Obviously, we don't want to take, 10 blood samples from an individual that's really sick right after they're dosed with the concentration to understand the PK. But we can get some blood samples from them during the course of the study. So it's very sparse. So I can't actually estimate for that individual their AUC or their Cmax. But what I can do is basically use mixed effect modeling, nonlinear mixed effect modeling, to try to model this thing. If you notice, this is the exact equation that I just talked about, but I've added random error terms. So for Ka, notice there's Ka times the exponential of eta 1i, which is normally distributed. So basically, I'm making an assumption that Ka comes from a lot of different individuals, and Ka has a log normal distribution, right? I'm doing the same thing with volume. I'm doing the same thing with clearance, right? I can do all of that and write that into a giant model. It's complicated, but it's solvable. And I can assume that these etas have a normal distribution, a joint normal distribution, right, with mean 0 and various variances. And I can assume the epsilon ij is normally distributed as well. Now I can, if I have data, I can estimate Ka, that is the average Ka, or the average clearance, or their average volume of distribution. But I can do even more. Now suppose you want to examine if clearance is related to body weight. I could replace in this wherever you see clearance, with clearance is equal to alpha times weight times body weight. So when weight raised to the beta. So when beta is equal to 0, there is no effect on clearance from body weight. I can estimate beta then, and basically have a sort of a feeling about what the effect, what body weight does to clearance, right? This is how observationally I can use the information from pharmacokinetics, the sparse sampling, to see, for instance, whether body weight, gender, other things that I have plenty of information of have an effect on the PK, on the clearance, on the AUC, if that makes any sense. Next slide. Okay. This is complicated mathematics and a lot of hand waving, and I know that, right? I've shown you that it's conceivable to use mechanistic models along with nonlinear mixed effect models to examine observational PK data. The question which you will see from the next section is that we can use the same logic for not just pharmacokinetic questions, but pharmacodynamic questions as well. Pharmacodynamics is not what the body does with the drug, but what the drug does with the body. Pharmacodynamics is what, oh, yeah, exactly. And to continue along this path, Oliver's going to talk for a while.
You can unmute yourself, Oliver.
Yes, now it works. Thank you. So as Brian has discussed, so pharmacokinetics is really what the body does to the drug. So the processes that Brian described, how the body absorbs, distributes, and metabolizes or excretes the drug. That is PK or pharmacokinetics. Here we're going to focus more about the PD part, and which is really the question of what is the drug doing to the body, and this is essentially the effects that the drug is leading to in the body. So you can think of this like a kind of causal chain here. And so if you look at the blue boxes, so the dose that you administer to a patient, let's say via some pills or via some injection, This leads to exposure or concentration of the drug over time. And the process between or relating dose to exposure, that is PK, that's what Brian has talked about. And this leads to an exposure or concentration profile over time, and which is depicted here at the bottom little graphic, which is so typically for a pill or an injection, leads to a concentration curve that raises for a while, and this is while the drug is still being absorbed. And then after some time, really the elimination of the drug from the body dominates, and the concentration will go less and less and less until the drug is fully eliminated from the body. Then in turn, this drug concentration or exposure over time will also lead to an effect or response over time. And just as an example here, so in the right-hand side, the picture, so here you see a patient with psoriasis, which is an autoimmune skin disorder. And so they have like red scaling itchy skin patches. And essentially, like for a drug to treat psoriasis, what you hope the drug to do is to make these skin lesions smaller and essentially go away over time with treatment. And so you could then quantify response by measuring how these skin lesions get less thick and essentially shrink away and go away over time. Understanding this causal chain of how drug dosing leads to drug exposure, and then drug exposure leads to response is really key in making decisions about what dose to give, how to treat patients in order to give the desired response at the end. And of course, as mentioned by Brian before, and I think it was also a question in the chat a bit earlier today, this relation is not the same in every patient, but is influenced by patient characteristics. So this relationship might be influenced by body weight, ethnicity, might be influenced by disease severity, or also other external factors like food effect and others. And so similarly, like for PK, as Brian has explained, this can also be taken into account for the PD part of this relationship. So trying to understand how is this relation similar or different between patients, and what factors might influence that relationship between exposure and response. Now, so as we've done for PK, we're really interested in the profiles and kind of development of these curves over time. And so likewise for the response, so for the PD part, we're also here interested in how do responses to drugs develop over time. And here we've kind of briefly characterized so different types of response patterns that are seen over time. So some drugs lead to a fairly direct or immediate effect. And so here you can think, for example, of anesthesia, where usually when the anesthesia sets in, it really within seconds or like minutes essentially takes the consciousness away. So it's the time span between giving the drug and then the effect setting in is really minimal. Likewise, inhalators for treating acute asthma, for example, also have time to respond really within usually just a few minutes. And on the right-hand side, you see a graph of a concentration time curve versus time, concentration versus time. And like in the top right panel, effect over time. And what you see here, and this is really what characterizes a direct response, is that essentially the peak of the concentration time curve happens at the same time as the peak of the effect time curve. So because of the really immediate nature or direct nature of the response, both of them peak at the same time. In contrast, some drugs lead only to a delayed effect. And you can think here of, for example, pain medication, which often takes like half an hour or even longer to lead to the desired response. Or some other drugs, like for psoriasis drugs, it can, for example, take sometimes weeks or even months until the desired effect sets in. And this you can see here in on the right-hand side in the bottom two panels. Again, concentration versus time and effect versus time curves. And what's different here in these two lower row panels versus the upper two panels is that the time of the peak concentration is not the time of the peak response or peak effect. So while here the concentration peaks fairly early on, it does take a lot longer until the effect has really reached its maximum point. Please note that these two, so direct and delayed responses or onset of drug effect, that these are really just two examples. And some drugs can lead to actually quite more complicated patterns of response and response over time. So for example, there are some drugs that can lead to drug tolerance, where you would, for example, see an onset of effect, but then the effect at some point starts going away, not because the drug goes away, but the body doesn't respond in the same way anymore to the drug. Similar to that, development of drug resistance. So if you have, let's say, a virus or a parasite being treated with a certain medicine, that pathogen can develop resistance to that medicine and can then just become like less responsive to that treatment. So like starting with the simplest case, so for a direct response like type PD effect. Like one way to describe that is using an Emax model or an Emax function. And so essentially what you see here is, so again, on the y-axis you have response or effect, on the x-axis you have drug concentration, and then you're describing the relation between response and drug concentration by these sigmoid type curves. And so essentially what you see is, so it's a monotonically increasing curve, it's a smooth curve. It's starting at 0. It's reaching an asymptote at 1, so which is kind of a relative effect, like the full response effect. And what you see here, and that's the 3 different curves, 3 different colors. Here are 3 examples of different steepnesses of the curves. So you have here the like alpha equals 1, the black curve, which is a fairly shallow curve, so it takes quite a while to increase. And then with the blue and the green curve, so green, for example, is a very steep curve where you have, like for concentrations below a certain threshold, pretty much nothing is happening. But then as soon as it's getting closer to this inflection point, essentially then patients almost immediately jump to full response. And then for all drug concentrations higher than here in this example, like let's say 20 or 25, would get to almost full response. Looking at this mathematically, so here on the left-hand side, you can see the mathematical formula for such an Emax model. And so what you see here is, so Emax is, so that's the maximum asymptotic effect that can be reached, which you can think of, it's like just a multiplier of this like response of 1 here. So like 1 is the full response, and Emax is just then the kind of numerical multiplier to whatever scale you're using to measure the effect. The x is, that's the concentration. So that's here, what's on the x-axis, the drug concentration. The EC50, that is the concentration at half the maximum effect. So here in this case, it's at 20. And so you can here read off on the y-axis, like 0.5. And the concentration that gives you 0.5 of the effect or half of maximum effect is here at 20 for actually all the 3 curves, the green, the blue, and the black curve. And the alpha occurs here as well. So that is sometimes called a steepness factor or hill factor. And this, as I briefly mentioned before, this is determining the steepness of the curve. And so here with alpha equals 1, getting the rather slowly increasing black curve, to then with alpha equals 2, the blue curve, and alpha equals 5, the green curve, which essentially almost immediately switches from no effect to full effect around a concentration around the EC50, which is here the inflection point where it's essentially making this jump. Now, before going to other types of PD models, I just want to briefly kind of do a very quick one-slide detour about analytical solutions versus ordinary differential equations. And as you might remember, Brian has before shown what's here on the left-hand side, this closed form solution model for pharmacokinetics of a one compartment model with like absorption from a drug, like single order absorption. And it's essentially this formula that Brian had already shown before. What you see on the right-hand side is exactly equivalent to what's on the left-hand side, but it's essentially like another formalism or another way of showing exactly the same type of model. And so here at the top, you see a schematic depiction of how you can describe this process, where here you start with a dose. This is then absorbed by a first order process into the systemic circulation that can be like blood or plasma. And then again, there's another first order process, which is the elimination, which is here denoted by this elimination rate constant K. And to describe this conceptual model mathematically, one can write this as a set of differential equations. And essentially what these differential equations do is really just formalize essentially all the errors you have here above. So here you have the dA1 dt, which is the change of drug amount in the first compartment over time, which is here in this kind of dose compartment, which is minus Ka, that's the rate which goes out of this compartment, times A1, which is the amount in that compartment to begin with. And then the second equation, which is Ka times A1, which is what is absorbed over time, minus K times A2, which is what is eliminated over time. And then you essentially just need to normalize the drug amount in this plasma compartment, dividing it by the volume of distribution, and that gives you the concentration. And so this is exactly the same thing then on the left-hand side, only expressed as a differential equation instead of a closed form solution. The pros and cons of the left-hand side versus right-hand side is, so the left-hand side is much easier to compute. So you can very easily with a pocket calculator just calculate that. Whereas on the right-hand side, you essentially need to do numerical integration to solve this differential equation. The advantages of the right-hand side is you can quite easily see how close the conceptual graph and the differential equation actually are. And it's quite straightforward to translate the conceptual graph to the differential equation. Also here in the left-hand side, this is still not very complex, but if you put a small error in here, mix up a minus or change the order of two expressions, it's actually not so easy to see that. So you need to be quite familiar with these models to spot that, versus on the right-hand side, it's a lot easier to see if something is actually off. The third advantage of these models on the right-hand side is that you can actually quite easily extend them. So if you, so here it's a very simple model just with these two compartments, but if you want to extend that, you can essentially just conceptually extend it here in the graphic, and then also here in the differential equations, essentially just add either terms to the equations or another equation. And here, this is just taking this one step further and essentially doing this addition to the simple compartment model for PK that I've shown before, and extending this to a so-called turnover model. And what this does here is it doesn't track just amounts of drug over time, but it also tracks here this R, which is a response or effect over time. And so if you look here on the right-hand side at this conceptual model here, what this essentially states is that there are kind of two processes. There's an inflow into the kind of disease state, and there's an outflow or improving of the disease state. And here, and just in this example, we're assuming that the drug essentially stops or inhibits the inflow, so the worsening of the disease. And at the bottom, then you see an example how this might look like if you kind of mathematically calculate what such a model would describe. So the kind of time pattern such a model describes. And this is what is here at the bottom. Again, concentration versus time curve. And on the right-hand side, a disease or R versus time curve, where as the concentration goes up, you do see the disease going away. But then after some time, after concentration is actually gone for quite a bit, you see the disease coming back. And so you see this like somewhat decoupled and somewhat delayed response of kind of like slow improvement of the disease, but then also after a while, and when there's really no drug left whatsoever, then you see also the disease coming back. And this is exactly the type of patterns that we see in quite many diseases, and these models are very useful for describing these relationships and these patterns. So let me just jump over that. So here, and really this is just to give you the kind of very high level idea that essentially these models, they can be extended and expanded in like many different directions. And just to give you a sense for the complexity here. So like this is a example of a fairly complex model. This is a model of myelosuppression. So this is a suppression of essentially blood cells due to chemotherapy. So patients that undergo chemotherapy for cancer, they like lose blood cells over time. And this, just to give an example of how this can be described, these like different transit compartments where essentially different blood cells are kind of transforming and growing into different types of blood cells. And then you have a feedback loop here. Here you have this proliferation loop, etc. So it's a fairly complex kind of process, but with exactly the same mathematical toolbox that I've shown before, you can describe such a fairly complex model with a set of differential equations where if you actually read the differential equations, even if you're not very familiar with differential equations, you can quite easily see how these equations really relate to the graphical depiction above. Just to give you a kind of broader idea of the kind of spectrum of models that one can use for different questions and circumstances. So I'm often thinking of this as a kind of spectrum ranging from very empirical models through semi-mechanistic models to quite mechanistic models. And empirical, you would have models like simple linear models, spline models, than the Emacs type models that we've already looked at today, then like slightly more complex models like the indirect response models, then the quite a bit again more complex myelosuppression model, but this can even still get more complicated and like fully mechanistic models. They're so-called physiologically based PK models, which are trying to describe PK essentially in all the major organs related to PK or where PK is of interest. And there are also these so-called quantitative systems pharmacology models where you are essentially trying to mechanistically describe how like drug and disease interact in various parts of the body and like affect various other like markers and substances that you might be able to measure, and thereby get a sense for how disease and how drug treatment affects these biomarkers. So what can you do with these kind of models? It's actually quite a like flexible and versatile tool. And so here, like in this NIAC 2018 publication, we've collected a couple of case examples, like what different people in different pharmaceutical companies, what they've done with these types of models. And just to mention a few examples. So these models, for example, can be used to help selecting the dose or dosing regimen for pivotal trials. So like before going into phase 3, you can use such a model to really pick the best dose to go forward into these trials. It can be used to reduce the trial burden for vulnerable populations because maybe you can test or study in fewer patients because you can use such a model to show that you actually kind of understand what to expect in such a population. It can help simplify dosing, which is of course good for patients, but also for healthcare providers. And there are a lot more things, like you can kind of probe what if scenarios, like, okay, what would happen if I would give the drug less often? What would happen if I change the dosing? What would happen if I do kind of like treatment on, treatment off cycling? You can use it to help designing the next study or integrate data across different studies. So it's really a kind of, multiple uses of these kind of mathematical tools. So now here in this part, I've really focused on kind of trying to look at this dose exposure response relationship. And the next part, Brian will look specifically at dose response studies, which is essentially just taking the kind of left-hand side and the right-hand side and saying that, well, it's all good and important to understand exposure and how exposure mediates whatever happens between dose and response. But for some goals, it's actually sufficient to really only understand what happens if you change the dose and how does that change the response, the clinical response that we're interested in.
Go back 2 slides real quick for me. I want to just say something real quick. No, one more. Back one more. I want y'all, no, forward one, sorry. No, where you had the Emacs model. Keep going. Good. Later in your presentation. No, not there. You're one graph. I'll tell you when to stop. Hit, stop. Go back. Here. I want you all to notice the Emacs. He says dose response down there. What's really interesting about the dose response relationship with the Emacs model, there's one parameter in that model, the Emacs, which if it's 0, means that the drug isn't having an effect, which you can do a test on. So one of the ways that you can get rid of multiplicity when you have multiple doses, if you have a model that works, is by changing the problem into that it's just being one parameter. That reminded me from Nee's presentation. So just to kind of keep in mind that in essence, we always should be looking, even for things like multiplicity, we can use some of the models, the simpler models that Oliver talked about, to solve really important statistical questions. Now you can go to mine. Next slide. Okay, so Oliver's just shown you all the things that we can use. We really wanted to get to the point where we got to the end of 2B where we have a dose. So I kind of told you what we do after we already have a dose with clinical pharmacology studies. Now, how do we determine what that one dose is to go into phase 3? Hopefully we know at this point how frequent to dose the drug, BI twice a day, once a month, a safe maximum dose, most maximum tolerated dose. Our goal is to get to one dose. Next slide. Selecting the right dose obviously is essential. If it's too low, we got a suboptimal efficacy. If it's too high, we're causing safety, right? And you know, there are all kinds of difficulties in getting that, but for time, let's get to the next slide. The thing is, dose responses look like that Emacs model that Oliver just showed. Here's real life example. It looks like this very often. Next one. So how would you pick the right dose here, right? Well, we want the one that delivers near maximal effect, right? But that's the one that's the lowest, because the lowest one's going to be the safest, if that makes any sense. Next slide. Conceptually, why do we have that S-shaped curve over and over again, even in this dose response? I mean, this goes back from the very beginning. If you remember right, I said there are receptors around. I deliver drug, right? The more drug I deliver, the more receptors are bound. Guess what? I can only bind up 100% of the receptors. So it has to plateau like this, right? As far as potency is concerned. But if that's really affecting disease, next slide. So I just want to talk to you about a particular type of drug, not because this is an important type of drug, but just to give you a flavor for what I'm going to say next. PCSK9 is a substance that you have in your body. It binds to the receptor for the LDL cholesterol. By binding to the receptor, there are less receptors for LDL cholesterol. Thus, the more PCSK9, the more circulating LDL, which causes, in the long run, cardiovascular events. This implies a PCSK9 inhibitor would reduce the amount of PCSK9, which reduces the amount of PCSK9, would also decrease the amount of LDL. This has actually been shown, right? But as you can see, there's a chain of endpoints that one can measure. Next slide. The amount of PCSK9, as I increase the amount of PCSK9 inhibitor concentration increases. The amount of PCSK9 decreases, the amount of LDL receptor increases, the amount of LDL decreases, the amount of arterial plaque decreases, the risk of heart attack decreases, and life expectancy increases. And here's the thing, next slide. They all have an S-shaped curve. And they all have an S-shaped curve basically because at the beginning, it has an S-shaped curve, right? Now, this is a generalization, right? You can find exceptions to this, right? Oliver explained that when I was taking time into consideration, I couldn't match those things up one-to-one. I'm talking about a situation where I just have a measurement at the same time. I'm going to generally have this S-shaped curve. This is important for us to understand. Next slide, right? U-shaped dose responses do happen. Here's some examples from some toxicology studies. Next slide, however. There are two main reasons for this. The main one is sometimes the body fights back. It ramps up the production of the thing you're trying to get rid of, right? In that situation, you probably don't have a drug. Probably. Sometimes the endpoint itself is not just measuring the positive effect of the drug, but the negative effect as well. Think of time until death as an endpoint. If the drug also causes serious adverse events, then the positive effect of the drug might be canceled by the negative effect of the adverse events. All right, next slide. So the key here, though, is to use the appropriate methodology. If we know we have an S-shaped curve, use an S-shaped curve. There's some more things that we can do about that look at different types of S-shaped curves and so forth and so on. But instead of pairwise comparisons to placebo, model it. And that's the key with dose response. Once you show that the drug works, then estimate a dose that produces near maximal effect that's as low as you can possibly be. Next slide. And this is how you would do that. Next slide. Okay, use the appropriate dose methodologies. Basically, dose changes response. Response changes, dose changes response. That's used in these dose finding studies. However, with that said, and Oliver can contest, sometimes the simple model, dose to response, isn't good enough. In the pharmacokinetic modeling that he just talked about, becomes very, very useful of understanding how to dose the medication. Next slide. Were you supposed to take this one over, Oliver?
Should I take it? Okay, sure. So this is Justin, this is really coming very close to the end, emphasizing the importance of informative study designs. And here in particular, the question, like which doses or how many doses to study in your phase 2b dose finding study. And so if you think of like these different designs where the black curve is the true, but at this point in time, like unknown dose response curve. And the red dots are the doses and the corresponding responses that you are testing and studying within your study. What is the kind of pros and cons, or what are the downsides of the different examples here? So if you look at the top left panel, here the issue, and so to just kind of describe that, essentially you have one low dose with pretty much no response. And then you have two doses with like very similar response, which are both at the plateau of the dose response curve. If this happens to be the doses that you're studying in your dose finding design, the problem is that you haven't learned anything about where the dose response curve increases. So you will have no way of arguing that not a lower dose than what you've tested here actually might be efficacious as well. And if you look at the top right example, so here again, you have 3 picked doses, but then you haven't actually picked a dose that is lying here on the plateau. So here, if you end up having kind of sampled or probed these 3 dots on your curve, you actually don't really know what's the maximum response if you've only observed response for these 3 like red denoted doses. Here at the bottom left, it already starts looking much more sensible. So you do have the steep part characterized quite well. So here by these actually 2 doses on the increasing part of the curve. And then you also have here a dose which is pretty much on the plateau of the curve. So you do have a good sense for where the curve increases, how steeply it increases, and then also what a maximum or close to maximum effect might be. If you look at the bottom right, this is even like maybe even better. So you have even a few more doses to characterize the increasing part of the curve. And now this is an important bit. It's essentially hedging against uncertainty of the location of the curve. And so when we're discussing that, it's important to just again emphasize at the time of the design of the phase 2b study, you actually do not have the black curve. So you do not know where the true black curve sits. So you can only kind of assume and anticipate. And then based on that, you place the red dots and try to essentially do these observations in your study. And if you're unlucky, you kind of misplace them in the sense that you either put them on the plateau or you put them all only on the increasing part, and then don't get enough information on the plateau. So really a lesson from that, or conclusion from that is, so explore really just like not just 1 or 2 or 3 doses in such a study, but the recommendation is really to do, like consider maybe doing 4 or even more dose groups, and also probe like large enough dose range. So recommendation is to at least go over at least 10-fold dose range, and not just probe, let's say, like 1, 2, and 3 milligram, but really probe over a wide enough range so that you actually have a chance of capturing a reasonable part of the dose response curve. Sometimes adaptive designs can help. So this is in particular when there's a lot of uncertainty up front, kind of how this black curve might look like and where this curve might be located. It can make sense to do adaptive designs, which, for example, if you look here at the top left panel, and if this is what you've observed, so these 3 red dots, then an adaptive design might then allow you to, for example, just put in like another arm, which then could explore further lower doses to then continue to characterize the increasing part, which you've missed with these 3 doses to begin with. Or likewise in the top right part, where you've missed to explore the plateau, and adaptive design could help you to then kind of iterate and add more doses, in this case here higher doses, to really explore where the plateau sits. Okay, so this now concludes our session, and really just to kind of quickly summarize and wrap this up. So at the beginning, we talked a bit about going from a molecule to a drug that we know how to use, where we've figured out the label and the prescribing information, knowing how to use it in patients. Brian has talked about the mechanistic principles of how drugs work, and here, so here in the yellow and cyan colored example of binding, that most or even like practically all drugs work by binding to their target, and that this is really the kind of most basic principle of drug mechanisms and drug action, binding of drugs to the target. Brian has talked about pharmacokinetics, about the ADME process, about concentration curves, how concentration curves look like, how you can summarize them, like Cmax concentrations, AUC, so area under these concentration curves, and how you can mathematically analyze them. We looked at the dose exposure response relationship and so PK/PD, how this causal chain is important to understand. We looked at different types of exposure response relationships. So here, for example, the direct response where the peaks are at the same point in time versus delayed onset of drug effect where the peak of concentration is happening at a different time point where the peak of the effect curve. And Brian has talked about the dose response characterization and essentially what you can do in terms of study designs and in terms of analysis methodology to properly characterize the dose response relationship, which then allows to select a dose to go forward into the large phase 3 type studies. And that brings us to the end of this presentation. And I guess then we have time for a few more questions.
Thank you very much. Can we please give a very big round of applause to two scholars that just concluded their presentations, Dr. Smith and Dr. Sander. From the online participants, I will please appreciate these two giant presenters, please. Online participants. Thank you very much. We really appreciate you for this beautiful presentation. This is one of the key areas in dose response methodology, pharmacokinetics and pharmacodynamics. They have treated us into a very deep overview of how to go into the business of pharmacokinetics modeling towards drug development. On this note, I want to call anyone that is interested, that has one or two questions to ask our resource persons in this area. I want to assure you, sir, that we have a number of our participants that are researching in this area. And that makes this workshop so relevant to what is going on within the research circle among our participants. And of course, not only in Nigeria, even outside the shore of Nigeria. So questions, please, from within the institutional room here and online. I want to entertain as much questions as you have before we round it up. Thank you very much. Questions, please.
Okay. Thank you, Oliver and Brian. Thanks for that presentation. My name is Femi Oyamaki. I just have a quick one. The start of your presentation, you talked about body tolerance. Body tolerances. I'd like to ask, what other attributes do we need to concentrate on when we're looking at the effect of this tolerance in the development of drug? That's number one. The second part is looking at the complexity that is that's associated with indirect assays. Okay, what would you advise? Because I saw some of the charts produced, the logarithmic one, and most people that are looking at assays wants to stay at direct assays. Okay, they want to assume that drug is not a function of the body tolerances. They just want to look at the relative potencies directly and look at how well they can characterize it and look at the interval, the credible interval. So what do you have to say to this? Then finally, talking about pharmacokinetics and pharmacodynamics, I believe that because of the complexity in terms of the expected discipline to perform optimally, what would you advise as experts in terms of developing curriculum in this area? Should it still continue to be exclusively for those that are in pharmacology or those that are in pharmacy, or that we should start having an hybrid of pharmacy students or pharmaceutical courses, statistics courses, and mathematical courses, such that we start having hybrid of pharmacologists that will be able to deliver on drug development. Thank you.
Oliver, why don't you try the last question?
Okay, so I can, I think, take on the first question on tolerance development, and the last question about like whether it should be restricted to pharmacology and pharmacy students. So regarding the first point on tolerance, and I mean, like this is a like fairly complicated topic, and also really depends a lot on the specific circumstances and the drugs you're looking at. But so if you think of let's say caffeine intake, I mean that's a classic example where the response to caffeine like typically has some tolerance development. So if you're not used to coffee and drink like a large cup of coffee, this makes you very alert and kind of very awake, and you might even have trouble sleeping for a bit after that big cup of coffee when you're not used to drinking coffee. If you're a regular coffee drinker, your response to coffee is still there, so you still do respond to coffee. It still does make you somewhat like awake, but the response is a lot less compared to someone who doesn't regularly drink coffee. And so this type of tolerance development, that is, and that's really drug specific, whether that needs to be considered for a certain type of drug or not. And it's, I mean, sometimes you can kind of argue, or you're anticipating based on the mechanism that some form of resistance or like tolerance might develop for that treatment. In other cases, it's something you observe empirically. So you kind of notice that the effect is going down over time, despite you keep up the exposure to the drug at a constant level, but still the effect somewhat goes down. And then in those cases, you need to try to understand and try to quantify like how fast does that effect go down? How quickly does that tolerance develop? You might be interested in questions like, does that tolerance go away again? So like if you take a break with coffee drinking, for example, you will be more alert again if you start drinking like your next cup after a 1 week break of coffee. And these might be questions that you also need to figure out for a drug if you've notice that there is tolerance development for that treatment. For the last question regarding kind of restricting to pharmacology and pharmacy students, I mean, and I think Brian and myself are maybe good examples of, or kind of counter examples, and like me as well. I mean, we're all like drug developers and all work in drug development, and all come from quite different backgrounds, and I mean, geographically, culturally, but also in our education. I think we all have somewhat different backgrounds. So my background, for example, is computer science. And I then, like over time, moved into computational biology, and then got more interested in pharmacometrics and application of quantitative methods in the pharmaceutical industry. And so, in my, and I guess my kind of own experience, like there are companies and there are kind of areas that are quite open to people coming from different backgrounds and bringing in different strengths. But of course, it always also does require that people keep learning and extend their horizon and do pick up like pharmacology and some like pharmacy background on the side if you're coming from a somewhat different background.
So the second question was about potency, if I remember right, and I think you've asked the question before, and you obviously were not getting your getting an answer that makes sense. I'm wondering if you take my email and just explain it to me carefully, because I don't get it right now what you're asking, but I know we're running out of time, so there's no reason to delve into it. But why don't you send me a note? And if I can't figure it out, I'll find someone that will, if that makes any sense.
Okay, that will be done. Yes. That will be done.
Okay. Yeah.
Okay. Yes. Okay, maybe just a quick one again. Okay. Dr. Adedi described the phases, explicitly. Now, the question I want to ask is, how does feedback, now when the drug is out there, how does feedback impact on the phases again? I'm talking about a backward movement. How do we now have the feedback impacting on the phases, informing a new knowledge that will contribute into phase 1 back through the phases that we have?
Great question. I think we're even to the point now at Novartis where we're having large phase 3 studies, and those are helping to inform our preclinical scientists about new mechanisms to undertake. And we've seen that, right? So we have that feedback mechanism throughout. I think we learn. So feedback as far as particular drug is concerned, that's one thing. Feedback about how we do our job better, that's something else. Or ideas about other targets and other places to go. These are things that we do. Oliver, would you like to add any more to that?
No, but I agree with what he said. And,
but if you think about the incredible amount of data that we acquire over time, the fact we've not actually in the past been very good of turning that information into knowledge for feedback. We're better at that now. And so we're hoping to see some wonderful things happen, but we've already started seeing promising things. I don't know if that makes any sense or not. So I can't get into details, but it's coming. We are very interested in feedback. We're especially interested in exploiting all this data that we collect for new things.
Yeah, thank you very much. Once again, I want to thank our facilitators, our resource persons from Novartis Pharma AG for these beautiful presentations that have been given us this afternoon. For those that have joined us lately, this is the third pre-conference workshop of the Professional Statistical Society of Nigeria, coming to you live from Nigeria, from the Nigeria Institute of Medical Research. And I have the honor and privilege to announce to you that this workshop is being proudly supported by Novartis Group. And then, of course, the 6th International Conference of this Noble Society, starting from tomorrow, is again proudly sponsored and supported by Novartis. A very big round of applause to Novartis, please. I want to appreciate our resource person, starting in no particular order, from Dr. Adeni Adewale. Very big round of applause to Dr. Brian Smith. Very active since we started. And of course, Dr. Sander. They have tried very, very well this morning. And I want to believe that we all appreciate your efforts, sir, for giving us these beautiful presentations. On this note, I want reactions from our participants. I want our participants to react to this presentation. From online, I want a volunteer to react from online, yeah. And from the institutional room here too, I want the reaction from us. And let me say this, and like I said in the morning, this presentation is being streamed live on YouTube. If there, by any means, for whatever reasons, you miss out on one of the sessions, you can download the video of these presentations and update yourself on what has been discussed this morning. I want to call on Dr. Anthony Ekpo to place the hat. Before I could call Dr. Domboso. Dr. Domboso is connecting with us from Italy. You
yes,
are
sir.
highly welcome. Dr. Anthony Ekpo, please. You have the floor. Your reaction, please. Dr. Ekpo, go ahead, please. You have some challenges. Okay. While you are solving that, let's call on Dr. Chris Domboso, joining now from Italy.
All right, sir. Thank you so much. Greetings from Italy here. I want to so much appreciate the presenters. It has been so wonderful and it has been very engaging from the time they started. I want to believe that this has added to our knowledge, and we, of course, many people that are working in those areas are going to really have something to take home and do better on their researches. So once again, thank you so much, Dr. Sander, Dr. Smith, and Dr. Adewale. And we want to believe that this is not going to be the last time that you are going to be with us in this society. We would love to have you on as you share your knowledge with us. Thank you so much.
Chris Domboso, joining us from ICTP in Italy. I want to call on Dr. Anthony Ekpo. Are you ready?
Yes,
are you ready?
Yes, Prof. Yes, I am now.
Okay, go ahead, please.
Sorry for the
Go ahead, we're hearing you.
I want to join my voice to say a very big thank you to all the presenters. I want to say that your labor to have given out this knowledge is not in vain, because we have been able to gain a lot from what you have given so far. Very many thanks go to Dr. Denny especially, and then Dr. Bryans and Dr. Oliver for all that they have been able to discuss. And then just like Prof said earlier, this area is an area where most academics have interest in terms of research. And then what you have been able to share is not in a small way. I mean, it's going to go a long way to help us further on the research. So on behalf of all of those online, I want to say very big thank you, and then wish that we can have this more and more of the time. So thank you so much, and big thanks go to the organizers. We thank them for the efforts they have put in to make sure that this is a successful one. Thank you.
With us, and I want to give his remark. Please come over.
Thank you so much, Mr. Chairman. Thank you for organizing this conference. It's not been easy for me all the way from River State, but when I get to this place, the presentation was very educative and enlightening, and also the organizer. But on my way coming back, I was following the process on the Zoom, and it's really interesting. For those who are not here, we pray that God should grant them opportunity next time so that they will be here. Thank you so much, Mr. Chairman.
Thank you very much. Thank you. Before I call on the president of this beautiful and noble society to come and give the vote of thanks, let me announce to us that Tomorrow is a big day to our society. Tomorrow is the opening ceremony for the conference proper, the 6th International Conference of the Professional Statistician Society of Nigeria, the 6th one in the series. And we are expecting the giants, the timber and caliber of the society to grace the occasion. Notable among them is His Excellency, the Executive Governor of Lagos State, Mr. Babajide Sanwo-Olu, who is going to give the keynote address at this opening ceremony. And of course, the rector of this beautiful polytechnic, the Yaba College of Technology in Nigeria here, is going to give a keynote address. And of course, the Director General of Nigeria Institute of Medical Research, where we are linking up with you live there. So, Obatode Salako is going to give us keynote address as well tomorrow. After which we are going to have a lineup of lead paper presentations, and a good number of our lead paper presenters have. We have one from US, another one from UK, and of course one from Nigeria here. So tomorrow is an action-packed day for us, and we would love if any of our resource person here could join us to grace tomorrow's program. From wherever you are, you can link up with us, and we have the history of pioneering this kind of a thing in Nigeria during the COVID-19, when there was no direction of what to do. We set the pace in Nigeria on how to have a virtual conference that link up every other scholars all over the world. So on this note, again, let me reiterate this, that both the pre-conference workshop and of course the 6th International Conference of this Noble Society of this year is being proudly supported and sponsored by Novartis Group. And we are, on behalf of Council of PSSN, we are most grateful to the Novartis Group. On this note, I want to call on the president of this society, person of Professor Olarewaju Shitu, to come and give vote of thanks to our elder scholars. A very big round of applause for my president, please.
Thank you very much, powerful National Secretary. A lot of appreciation has been given to the speakers of the day, as well as the organizers of this pre-conference workshop. Nevertheless, I'd like to thank the speakers again. The first one is Dr. Adeni Adiwale, his presentation, Dr. Brian Smith, and Dr. Oliver Sanders. I was impressed by the presentation of Dr. Adeni Adiwale on a number of issues. And I was impressed and excited when somebody from my country, Nigeria, and particularly from Ladoke Akitola University, is up there making waves. We have enjoyed your presentation, just as we have enjoyed Dr. Brian Smith's presentation and Oliver Sanders. And this conference workshop was organized in order to further develop the capacities of, statistical capacities involved in the development of drugs to combat COVID-19 and some other diseases in Nigeria. And I want to say that this is in line with the goal of this society, that is the advancement of statistical theory and its applications. And the applications you can see from what the papers that are presented this afternoon. So once again, I would like to thank the council members of this noble society for their belief in us that at least we can make a difference in the Nigerian statistical community. I'd like to thank our numerous members of society that are not here, but are present online. Just as I would like to thank all our guest participants from all over the world for attending this pre-conference workshop. And I want to believe that many of us, including myself, have learned so many things from these presentations. I also like to appreciate Novartis Pharma NG for the support of $3,000 to a conference that is scheduled to start tomorrow and it will last till Thursday this week. Once again, I'd like to thank everyone for attending this conference workshop. Thank you very much.
Thank you very much. It has been an action-packed day for us, and you will agree with me that for all of us that have participated, either online or in person here today, we have updated ourselves in terms of knowledge by what we have been presented to us. Whichever area of statistics you are researching into, what was given to us today, we have one or two things to pick to update your knowledge. And then on this note, I want to call on Professor Ngo, who is a member of Board of Trustees of the Society, to give the closing remark. Thank you very much.
Thank you very much, the National Secretary, the National President of PSSN, and all other members present. I send my greetings. I send my greetings to those watchers from the internet. They have thanked our presenters, and I want to add my voice in thanking them. It benefited efficiently and eminently from what they presented to us. So I'm sure that we're going to take it on from there and develop further in those areas. Well, I will not forget to thank the technical crew that are in front of me. I think they have done marvelously well. And I always appreciate, when I see them press one or two buttons, I appreciate that because I know they are trying to make things to move, in the right direction. So on this note, I want to say tomorrow will be another day, and I'm sure tomorrow will also be a successful day like today. Thank you very much.
Thank you very much. So tomorrow activities we are going to start by 9 o'clock, God willing. I want us to please do join. For those that are joining virtually, you can be very sure that we are going to link up as early as 8:30 thereabout. For those that will be joining us physically, please let's meet at the venue to start the program in earnest. We have a lot of things to do tomorrow, and the parallel sessions for the paper presentations we commence in earnest on Wednesday, and we are going to waste no time. Those that we lay the parallel session have been duly informed, and the paper presenters too, they should be online on time so that we have a lot of, we have more than 100 papers to present. we have more than 100 papers to present. So many that we have more than 100 presenters. So from Wednesday and Thursday. So it is going to be a very busy day for us for those 2 days. So let us start early. So tomorrow and the other days of these our conference sessions. On this note, I thank every one of us for being part of this beautiful session this morning. Our people from our colleagues, our research colleagues from the universities, once again, on behalf of Council, we say a very big thank you to you for making it a day with us and for making this a success for us. You shall ever be remembered in the history of a professional statistician society of Nigeria, a notable professional statistical body duly registered in Nigeria. So we are going to partner with you more than this in the future. So our participants for the pre-conference of this year, you are going to have your certificate. Immediately after this conference, we are going to activate individual portal where you can download the ticket to access your certificate for participation at this conference. So at this pre-conference session. So that is another additional information I have for us. So maybe in the next 30 or 40 minutes, just go back to your portal and download your e-certificate. Thank you very much. See you tomorrow. Bye-bye.